Alô, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Boa tarde a todos. Meu nome é Adriana Bonamatos, eu estou aqui representando o Programa de Pós-Graduação em Dentística, que atualmente tem 5 áreas de concentração: dentística, biomateriais, biologia oral, endodontia e laser e odontologia. É um prazer ter vocês todos aqui, inclusive os colegas que estão nos assistindo pela transmissão do nosso canal do YouTube, e receber os professores da Universidade de Birmingham. Muito obrigada a Flávia Pires Rodrigues, por essa oportunidade de trazer os professores para apresentar as suas palestras e conferências e estreitar os laços de colaboração com a nossa instituição. E o nosso primeiro palestrante vai ser o professor Phil Thompson, que vai conversar com vocês sobre Adopting a Biological Approach to the Treatment of Pulp and Periradicular Disease, que apesar de ser uma palestra algo endodôntica, né, de alguma maneira, sempre tem interesses associados para todas as áreas do conhecimento. Eu estou muito feliz de ver esse anfiteatro cheio de gente, espero que tenha bastante gente online assistindo também. E agora eu vou passar a palavra para o professor Phil, and I would like to thank you for coming here to visit us. It's a pleasure to have you here, and I hope that we have a wonderful time together here, not only at FOUSP, but in Brazil. Thank you very much.
Thank you very much for everybody coming to listen to us this afternoon. We've had a fantastic time today in Sao Paulo, visiting your school, seeing what you do in terms of your research and your clinics. So we've really enjoyed this morning. It's been really good to see the breadth of what you do here, because we think it's a great school, wanting to engage with you. So we're really excited to be here. Thank you. I also say hello to those that are on YouTube in your sister schools. I don't know whether if there's a camera and I'm videoed at all, not entirely sure. So hello to you guys in your sister schools. Excellent, perfect timing. Thank you very much. We've all come from Birmingham, from the University of Birmingham, in the middle of the UK, effectively. It's a relatively small city in by Brazilian standards, as Flá described it. I'm sure lots of you will know Flá. She described it as a small, cute city. And I think that's perhaps sums it up. In comparison with Sao Paulo, we have a population of 1 million, and I believe yours is 27 million, so significantly different. We have a nice eclectic range of architecture in terms of modern and new. We were, or old and modern. We were quite an industrial city. And there's been huge developments in the last 20 or 30 years since the industry declined, and we're really blossoming now as a really vibrant city, and have super communications with London, and it's almost like a suburb of London. It's probably quicker to travel from London to Birmingham than it is across Sao Paulo, I think, in the rush hour from what we've seen. So our university is a beautiful traditional red brick university. It's probably the reddest brick university in the UK. And it's quite a traditional. We have a beautiful clock tower in the middle of it based on architecture from Florence. And you can probably see some of with our promotional banners represented here, the architecture from the university and the Birmingham thriving together. So in terms of our dental school, it's a brand new dental school. It's only been open 2 years, 50 million pounds building where all of our research, our teaching facilities are all integrated. We've got really high-tech facilities because all of it's practically brand new. So it's a fabulous working environment. So a little bit more about me. I'm the head of conservative dentistry and endodontics. I'm an academic, so I do clinical work, teaching and research. In terms of my clinical work, I run a service in endodontics, so we have more complex endodontics referred to the hospital that we treat ourselves and as part of a training program for our registrars, they're called in the UK in terms of specialist trainees. And we really have a great history in using the operating microscope. We've, through Phil Lumley's had the operating microscope for over 25 years, and we're really carrying out, hopefully, what's considered high-level endodontics. In terms of our teaching program, I'm in charge of the undergraduate program. We're developing our postgraduate taught and programs, which Josette will speak to you a little bit about, and we're also in the process of developing a bespoke endodontic program. And I'm going to talk about my research now, and it's all evolved around the area of endodontics. So I have a declaration of interest. Unfortunately, it's not a commercial one. I would like to say that I'm paid thousands of pounds by Dentsply, but unfortunately not. I'm a pulp lover, and that's my declaration of interest. We've got a great history with the pulp in Birmingham in terms of our research for many decades, through Professor Shovelton and Tony Smith. Tony Smith is a name you may have heard of, but we have a great background in terms of this. We even, if you look very closely on our new building, have 6-meter high odontoblasts as an artist impression of odontoblasts. That's how proud we are of it. So, being a pulp lover, it's important to understand why we are trying to preserve the pulp vitality, because it acts as a defense system to the tooth in order to protect itself. It serves as a little bit of a proprioceptive function. If a damaged tooth still isn't fully developed, then we want it to continue to develop and protect it. However good we are at endodontics with our fancy instruments and all our gadgets and our microscopes, we still can't do it perfectly. It's not always going to be successful. And because we get our big instruments in there and remove dentine, we're effectively weakening the teeth so they're prone to fracture. So one of our big areas of research, you'll hear about others, is a focus on trying to ensure that the pulp is safe during treatment, and if it does become damaged, how we can regenerate it effectively. And this is some of my work that I'll just mention in terms of the response of the dentine pulp complex to calcium silicate cements. This is, I'm sure you've seen this paper where Hal Duncan, who's a collaborator and friend of ours, he did part of his PhD with Paul Cooper, all his PhD, did some beautiful histological work with Ramner from Switzerland, and showed really very robustly and categorically that the calcium silicate cement response, or the pulp response to calcium silicate cements, is better than calcium hydroxide, and it's far more favourable. And we see these beautiful dentine bridge formation and tertiary dentinogenesis. So really, some of my work is trying to answer how we form that tertiary dentine in response to the calcium silicate cements. And we've got a great history at looking at growth factors, which I'll come on to. But calcium silicate cements and MTA, and there's lots of different ones out there now, interacts with the surrounding tissue fluid once it's set and as it's setting. And it's quite a complex material, and there's lots of different ions in there. And these ions get, some of them will be solubilized into the local tissue fluid. So actually you have with the material itself, it modifies the tissue fluid around it. And perhaps that might have an effect on the local tissues, whether it's the pulp or the dentine. There's lots of different ions in there. Josette, who will be speaking to you later, has published some research on this, and so have I as well. And we looked at the local interaction of dentine with these solutions. And if we look at the molecular structure of dentine in terms of the collagen and the different proteins that exist within the dentine, within the hydroxyapatite, there's sequestered or fossilized growth factors within that dentine. And as I was saying before, we've got a great history as regards analysis and understanding these growth factors that exist in dentine and also the pulp extracellular matrix. And we showed that this solution with that's quite complex, full of ions, that these growth factors can be released from the dentine. And it's looking at the effect of these growth factors potentially could kick off the reparative mechanisms of the pulp, and that's why the tertiary dentine is being formed. So in terms of doing in vitro work and trying to extract these dentine extracellular matrix components, it's quite a laborious job. It's a perfect job for a PhD student if you want to test their resolve and see how good they are, because you effectively have to make pure powdered dentine and lots of it to then be able to determine what growth factors are released by different materials and potentially different irrigants. And so we've got a great experience with solubilizing these growth factors from dentine and doing subsequent experiments on them. So we've shown that the growth factors from these clinically relevant pulp capping materials releases a number of different growth factors out there that could kick off the reparative mechanisms of the dentine pulp complex. And when we're looking at this, we look at, it's a process of wound healing. And I like cooking, and unfortunately, if you try and be like the TV chefs and chop, 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 you can evoke wounds. And I thought, I've got to try and make something out of this wound. So I thought I'd take some photographs over a period of time of it healing. And effectively, because of the wonderful body that we have, things heal, and an exposed pulp is quite similar to that. So in terms of the mechanisms that we're looking at, in terms of hemostatic phase, inflammatory phase, looking at the cellular responses to that, if we're trying to model this in vitro, these are the processes that we investigate. And we found with these extracellular matrix components that are released from the dentine with different clinically relevant materials, especially MTA, that you get increased pulp cell proliferation and chemotaxis. We discovered a growth factor within the dentine that hadn't been identified before, which was quite unusual, that we wasn't expecting, and we thought we'd investigate this a little bit closer, hepatocyte growth factor. And we showed that it is released by just MTA and not calcium hydroxide, so possibly that's a key one. And we showed these molecular processes in terms of mineralization and the response of the pulp cells, and differentiate into odontogenic-like cells based on PCR profiling, to sort of suggest that these growth factors do have an effect on the reparative mechanisms of the dental pulp. So, sort of investigating periodicular disease now, we know that it's caused by biofilm and microorganisms within that contaminate the root canal following necrosis of the pulp. And it was really interesting to hear about some of the work that you're doing on this here in the building and in your clinics with Erica today. And sort of assessing biofilm removal is really very difficult, but we know that it's very, very important because there's lots of recesses within the complex root canal system that exists that this biofilm can hide. And we know it's impossible to sterilize the root canal system. We do our best, but even when we can't find it, we do our best, like in this case of this lower premolar. If we disturb the balance, then it still might evoke healing, but that isn't a predictable way of doing it. So we're investigating biofilm removal as being a really key thing in terms of management of periodicular disease. when we actually do the investigations on this in vitro, actually doing it is difficult because of reproducing complex root canal systems can be difficult, and much of the research that is done in interview blocks, and it's not really representative to or natural teeth, and it's not really representative to the clinical situation because of the complexities of the root canal system and the way that we look at it. And what a lot of the methodologies approaches in vitro is to use extracted teeth and try and cut them in half and open them up and then inspect the internal surface of the root canal system. But by opening up these teeth, it's quite easy to contaminate the internal surface of the root canal system. So to overcome this challenge, we're trying to look at non-contact methods of investigating what's going on inside the tooth regarding cleaning. And Paul and Damien have done some work in terms of micro-CT analysis and shows that debris that gets built up during different instrumentation techniques. Interestingly, reciprocating techniques tend to build up lots of debris and push it rather than bringing it out of the root canal system. It pushes into the isthmus and all the aberrant root canal anatomy. But building on this work, myself and Damien has got a really good PhD student from Iraq, Firaz, who's developed with us an excellent in vitro model, which I'll come on to. He's also done some micro-CT analysis, and we have an in-house micro-CT which Paul Cooper has built up within the school and provided excellent support, and we have use across the university with our micro-CT facility. And in terms of the work that Firaz has done, he's shown that these new off-centered instruments remove debris. They create a lot of debris, but they tend to remove it better than the rotary instruments. So that's a really useful non-contact method of assessing the internal parts of the root canal system. Firaz has, with us, developed a nice model that's based on a micro-CT image of a natural tooth. Well, we can do any tooth really and reproduce any root canal system, but frequently use a mesial root of a lower molar with a continuous isthmus all the way down it. And we 3D print these now in 2 halves. The reason why we 3D print them in 2 halves because it represents the clinical situation really very well, but once we carry out an intervention, we can open them up without contaminating the internal surface. And it's a really useful system. So you can see the 2 halves, and then it sits in this cradle. And you can fill it full of biofilm and grow biofilm within the root canal, then put it together, assemble it, and carry out different interventions, whether it's instrumentation or irrigation, or there's obviously numerous different interventions you can carry out. Now this model is set up. It's been optimized to make sure that it's robust in terms of assessing the biofilm that grows within it, and determining how the biofilm sticks to this model to make sure that it's clinically representative using a relatively sophisticated technique called AFM, which I'm sure you're familiar with. Rather than sectioning and sampling of the internal surface of the root canal system, we can do a whole assessment using some nice image analysis techniques. And it's a really neat way of assessing different interventions of cleaning the root canal system. And Ove Peters has picked this up from some work that we presented at ESE last autumn, and he's on sabbatical at the moment in Brisbane and is using this model, which is, he was very impressed with it. We can also use another method of OCT from a collaboration that we've got with Luc van der Slauw in Groningen, assessing both biofilm and debris that builds up within the root canal system during preparation. This is effectively like ultrasound of the internal surface of the root canal. And using different image analysis, we can differentiate between the debris and the biofilm to assess throughout the whole canal how well it's removed effectively. And again, these off-centered instruments based on our work, ProTaper Next and Revo S, do work very well at this. We're also looking at therapeutic approaches to irrigation in terms of regenerative endodontic procedures, and using our background in terms of growth factor release from the dentine in order to enhance the internal surface of the dentine for revitalization and revascularization procedures. So we want to effectively prepare the dentine surface such that there's lots of growth factors on the surface that is going to allow a favorable response for the cells that are going to attach to the dentine during regenerative procedures. And we've got a nice in vitro model developed to assess this with one of our master's students. And looking at the sort of end of our endodontic procedures in terms of obturation, with the development of calcium silicate cements, we're looking more at a far more biological approach rather than just doing our best to fill this root canal system. Perhaps we can help induce healing of the periodicular tissues and try and improve our success rate for those cases that don't actually work and we respond well with our apical periodontitis to get some bone form instead of having that inflammatory tissue of the apical periodontitis. And it's this interface which is important between the foramen, the internal part of the root canal system, and the external periodicular tissues. And rather than put something in there that is going to aggravate those tissues, let's make sure we put something in there that's biocompatible and perhaps might even help with the healing process. So there are calcium silicate sealers. There's a number of them out. The way I like to categorize them in terms of synthetic ones and Portland cement-based sealers. And there's lots of them out there, and obviously the Angelus project, which I'm sure you're very familiar with, MTA Philipex. So I just thought I'd show you this little video we use as a teaching video in terms of continuous wave obturation. But what I want to show you, I'm less interested in the technique. When you do this down pack with a heated instrument, to watch the flow through the lateral canals of the sealer, go through the lateral canals there. So those sealers really need to be biocompatible because we can't always control them, and they do move outside of the root canal. And that's why I think calcium silicate sealers are really very interesting. And the ones that we've looked at, they tend... Sorry about the sound. They tend to flow less under heat. There's controversy whether we should be using them with heat or not, but more of the research will come out to see whether that's a good technique. Apologise for the sound. That was the radio in the background whilst I was recording the videos. That wasn't meant to be on, but hopefully you get the point. Okay, that's good. We've looked at some micro-CT analysis of this with the imaging techniques. We can basically find out the voids where we haven't got radiopaque material to assess the quality of these sealers. And we're looking actually at a single cone technique with the calcium silicate cement, which is quite interesting because that's a nice simple technique. If you can come up with a single cone technique rather than using all the fancy obturation gadgets that we have, then that's going to make it simpler for the everyday dentist that is going to be treating most of this disease. And we found out in this study that actually using a single cone technique with the calcium silicate sealer fills as much of the root canal system as those lateral condensation and continuous wave. Myself and Josette have recently got a ESE grant to look at this in more detail and do a clinical study with some in vitro work. So this is hopefully giving you an idea of the work that we're doing within endodontics at Birmingham. We're really lucky. We've got a highly motivated team and excellent facilities altogether at our university, and we're really keen on collaborating to make sure that we can answer these research questions for the benefit of our patients. So thank you very much. Thank you for having us.
Nós vamos guardar as perguntas no final para a gente não se atrase com o andamento das palestras, ok? Aqui, ó. Thank you, Dr. Paul Cooper, for staying with us.
Thank you very much. It's a real honor to be here, and this is my first trip to Brazil, and everybody's been really friendly. It's a little bit warm, but we're having a lovely time. Thank you very much for giving me this opportunity to speak. I'm going to say a few words about Birmingham and the research that we do, and a little bit about myself, but hopefully there's a bunch of stuff that you can take away from this just to see how the School of Dentistry in Birmingham operates. And as Phil says, we're extremely keen to be collaborating or to try and get collaborations off the ground. So Birmingham is there. It's in the centre of the country. Phil's already alluded to that. There's London. And we're famous for quite a lot of things. Curries. You can get a good curry in Birmingham. It's famous for chocolate, Cadbury's chocolate. There's some famous chocolate that they make, and obviously that's good for dentistry as well. So that keeps us all in a job. We can play football, or some teams can. They're not doing too well at the minute, Aston Villa, but they're coming back. So Birmingham's really good in the fact that it's centrally located, so you can get all the way around the UK, but also you can get good flights out of Birmingham to many places in Europe. You can get into London in about an hour and a half, maybe a little bit less. It's a lot cheaper than living in London, so Birmingham's an ideal place to come to. If you want any tips on living in Birmingham, coming from Brazil, Sibele was with us for 6 months, and excellent ambassador for your school, and she, everybody had a great time. It was a real pleasure to have her. She did some nice work. So the School of Dentistry, Phil's already shown the picture, brand new school. We've been there for a couple of years, and we are a global leader in dental research. Every few years, I think 6 or 7 years, the government wants to know how good research on the whole is working in the UK, and dental research is assessed to see how good it is. So there's a process called the Research Excellence Framework, and we're not the biggest school in the UK, but we were #1 for our internationally excellent research that we were doing in terms of publications, and also the impact that we have on society in terms of economy and things like translation of our technologies. So we've done extremely well. We're a good school to collaborate with. We have lots of international collaborations as well, and that's usually important because, as I'm sure you're aware, international publications with several authors from different countries actually have a greater impact, and the studies have actually shown that. So we do a lot of collaborations, and obviously, the more we can do with you guys, even better. You can look at the SciVal and Scopus data, and you can see that actually we're doing really well on that as well. The QS World Rankings, I was talking to Damien Walmsley earlier, and I think we're at 19 and you guys are 18. So, together we can go even higher, I think. So we want to keep building on this success, and this is a £50 million school. You can see the odontoblast that Phil's mentioned earlier, artistic impression, so maybe not wonderfully accurate, but this is what the school looks like, and we've got, it's brand new, nice and fresh. Brilliant teaching facilities, brilliant clinics, and labs as well. You can get this information if you just Google any of our names. I just say a few things about myself in that. So I'm Director of Research. I lead the oral biology teaching, and I'm Deputy Head of School. I'm not a dentist. My background degree in genetics. I've done postdocs in the States and in industry, and I came to dentistry in 2000, came to the dental school in Birmingham. I work across a variety of areas, including with Phil, Josette, Damien. I do pulp biology caries, stem cell repair, look at molecular and cellular aspects of periodontal disease with Ian Chappell, Mike Millward, and other researchers as well. Tissue engineering and biomaterials with Dick Shelton, Will Palin, micro-CT research. So I'll mention a little bit about the photobiomodulation research we do as a group, and I'm involved in some toothpaste abrasivity research as well. So there's a whole bunch of stuff that I'm involved in that the school's doing. So if any of you guys have got any aspirations to come to the UK and plug into some of these areas of research, and obviously get in touch. And as I say, you can get our information either by talking to us now or Googling and getting emails, and we're very happy to receive emails like that. So I was given this title, Healing and Inflammation in Dental and Pulp Complex. It's, as I say, we're visiting a few universities here. It's a bit of a whistle-stop tour, but we're really enjoying ourselves. This is one of the areas that we've got quite a critical mass of research in that I'm involved in. And the way I look at it is that dentistry is continuing to progress and move forward. And this was a few hundred years ago, some images that Damien Walmsley provided me with from the BDA. And you can see it wasn't too good in those days, and it's significantly improving now. And the biomaterials areas were all a little bit nastier. They were pulling teeth out and selling them. And you like to think, we continue plugging away at biological processes, understanding pathogenesis, understanding tissue repair processes. In the next 50, 100 years, all sorts of things ought to be possible in terms of maybe regrowing teeth, etc., or components of teeth. So, I think it's obviously the more we do, we might not think we're doing great amounts at the time, but I think it all builds up and contributes. So dental disease, caries leads to the progression, the breakdown of the dental tissues, breakdown enamel dentine, gets into the pulp, causes infection of the pulpal tissues. You can lose the pulpal tissue, you can get periapical abscesses. And obviously, it goes really without saying that World Oral Health Report 2003, this is a big problem across the globe. And even in developed countries, fillings, restorations aren't always that successful. And in fact, studies have shown from people like Trevor Burke in Birmingham that after 5 years, a lot of these are failing, retreatment costs, loss of teeth, etc. In the UK, while this is getting quite old, 16 million old data, 16 million restorative procedures costing around 4 billion per annum. And obviously, this kind of treatment is beyond that of developing countries. So if we can start to harness biological process to tissue repair and regeneration and protection, it's likely to be much better. So where we are, we've got obviously the infection on the tooth that Phil's kind of mentioned, and you've got enamel breakdown, dentine breakdown, infection of the pulp, periapical abscesses. And if we catch it at an early stage, you can do a drill and fill approach and restore the tooth, and hopefully that will survive, but it might not last too long, and it could lead to further complications. Root canal therapy if the infection's too bad throughout the pulp and it can't be saved, but at least you can save the tooth in a functional manner, remove the pulp and cap it off. cap the tooth off, and that may last as well, but obviously there's limitations with that as well. And it may be that this is unsalvageable, and the next approach is to put an implant in the bone. We do some titanium implant research as well, and put a crown on that. And that, as we know, can survive for a few years, but there are issues with that, and peri-implantitis can occur. And there's obviously a lot of research into that, how to increase the longevity of implants. So, as I say, future dentistry, whilst in the short term we need to be working with materials and getting the most out of materials, we need to understand the biological processes that are involved in repair, disease pathogenesis, etc. And if we do that, we're likely to have better outcomes for our patients. So that's where I'm coming at it from biological approaches, and that's the basis of my talk, essentially. So one of the simplest things to do really is to look across the literature and look at what's the where we're kind of started was how would the dental tissues recognize and respond to infection. And some of this is relatively straightforward, but at the time of doing this work, at the time of getting into it was a question of, well, we know that the enamel can be broken down by the bacteria. We know that the dentine's it's got tubules in it. The enamel can get further broken down, the dentine can get broken down, and you can get to an infected pulp. And if we look at this sequela of events, we're aware from other news, other diseases, that a whole bunch of toll-like receptors or receptors that can detect bacteria could be involved. So, and subsequently studies have shown that the odontoblasts in this histological section around the edge of the pulp, there's the dentine, the porous dentine with its tubules, the odontoblasts with the processes in the dentine, and then we've got core pulpal tissue. So initially, at an early stage, it will be the odontoblasts who express these toll-like receptors and can detect the infection. Subsequently, as the infection rages, the fibroblasts in the core, resident immune cells that are there as acting as sentinels can start detecting this infection. We even know neuronal cells express toll-like receptors, blood vessel cells, endothelial cells, and stem cells can all be involved. And there's a balance between how they can start combating this infection and maybe even stimulating repair responses. Or they generate a really chronic infection, inflammatory response in response to the infection. So, as I say, going back a few years, we had an excellent PhD student, Julian McLachlan, and we published this work in infection and immunity. We basically just asked the simple question: if we look in diseased teeth, do we see lots of inflammatory molecules? And this is just a PCR approach showing that in diseased pulp, you get lots of or high levels of classical cytokines and calcium signaling responsive molecules as well, S100 family molecules, and it's quite simply, and proteins are upregulated as well. So quite simply, you get an inflammatory response in response to the bacteria that are found on and within the tooth. Subsequently, following that, this is not our data, but it's kind of a replotting of data that just looked at the immune response in the pulp. So you stimulate the cytokines signal for an immune response, and so we've got a healthy pulp there. Once carious infection starts to occur, you get an influx of the immune cells in the pulpal tissue beneath the carious lesion, and that might sound relatively obvious, but it needs characterizing, it needs to be understood. And you can see from this plotting of data going all the way from no caries, you've got a few immune system cells within the pulp, but if you go to full pulpal exposure, you've got lots of immune cells. You've got a variety of immune cells in there that are trying to combat the infection that's occurring within the pulp. As I say, we published some of this a few years ago as well. So Phil's already alluded to the similarities between infection or tissue damage and how the tooth might repair as well. So if we've got an infected pulpal tissue or damaged pulpal tissue, wounded pulpal tissue, you've got the bacteria. The immune cells will start to detect this, the tissue resident cells, the fibroblasts, the structural cells, endothelial cells, etc. They'll start signaling, they'll start releasing cytokines that will then cause the trafficking of neutrophils into the pulpal tissue. The neutrophils and macrophages will come in and they'll start to try and break down or try and remove the infection. And hopefully, if these cells can do this, the immune response works well, and maybe due to clinical intervention as well, you can actually lead to hopefully repair of the tissue, settling down of the inflammatory response, and healing of the tissue as well. So we published this as a review article with this hypothesis in mind last year. There's a bit of a but going on here, because it's a rather big but as well. Not only have you got the inflammatory response raging, you want to try and repair the tissues, but cells such as the neutrophil come into the tissue, and whilst it's trying to combat the bacteria, so there's the neutrophil, and we'll see some bacteria coming in at this moment in time in this video, the neutrophils will release their DNA, and they'll do it in this kind of Spider-Man-like fashion. And what that's trying to do is limit the spread of the infection within the tissues. Now you think, well, that's great, and that's been shown in many diseases, that it's an to stop important process to stop bacterial spreading, because obviously we don't want to spread into other parts of the body. And we know it's induced by bacteria. Cytokines will stimulate neutrophils to do this. So, yeah, as I say, a bunch of stimuli are important for this, and it's likely via TLR mediation and also cytokine receptors, which is relatively complex. And it requires ROS production, the relaxation of the DNA, and other enzymes that are involved in the neutrophil. And you think, well, that's absolutely great, but and we know that bacteria, a whole bunch of dental bacteria that are involved in dental tissue infection, can stimulate net release. So this is just measuring DNA release following neutrophils being stimulated by a range of bacteria. We can see some SEM images here of bacteria and neutrophilic DNA that's been expulsed as nets, and the bacteria associated with the DNA. It's all due to electrostatic charges, and this is stopping the spread of the bacteria. And it's also has been shown that molecules such as the histones that the DNA is wrapped around can also kill some of these bacteria. So it's stopping the spread and hopefully limiting the infection of these bacteria. However, I say it's a bit of a but. The problem with this is that the release of DNA from the neutrophils is also seen by the host tissue. So we've taken pulp cell systems which contain dental pulp cells as well, and we've done viability assays, cytotoxicity assays, and what we can show is that actually the DNA in the form of nets is leading to death of these pulp cell populations, and if we look actually more closely, we can see that likely to be due to histones. So the body seeing this as damage signals, and it's actually inflammatory response is not only trying to fight the bacteria, but the body's seeing this, and it's killing cells, and it's also causing chronic inflammation as well. So whilst the tissue is being damaged by the bacterial infection, and due to the enzymes and toxins produced by the bacteria, the host response as well, whether it's the nets I've mentioned or the matrix metalloproteases, which enable the neutrophils to traffic, reactive oxygen species that the neutrophils are making as well, to try and kill the bacteria, you're getting a lot of tissue damage collaterally as well. So there's a lot going on in diseased tissue. In an ideal world, when you've got the inflammation, you'd like it to resolve so that repair can be stimulated or can be facilitated. So low-level inflammation, I'm not going to go into the studies, but there's quite a lot of evidence there that low-level inflammation enables repair. It actually facilitates it. So some of the signaling pathways for P38 MAP kinases can, whilst they're involved in inflammation, can also be involved in repair as well if the inflammation's not too severe and is not chronic. If the inflammation kind of goes chronic, there's too much infection, then the repair processes are going to be inhibited as well, and you're going to get a lot of tissue damage. So there's a few processes for the tooth to repair itself, and this is termed tertiary dentinogenesis. It's the tooth's natural wound healing response. It's like if you damage your bone, if you break a bone, it will try and repair itself. And the tooth is exactly the same. It will try and repair itself. However, the problem is it's a dirty environment. There's lots of infection there, but it wants to undertake this process, and it's a process called tertiary dentinogenesis. Tooth repair can occur in response to caries, abrasion, attrition, restorative placements. Is this a better one? Is this better? Yeah. Okay, thank you. So caries, abrasion, restorative placements can all stimulate tertiary tissue or tertiary dentinogenesis and tooth repair, and it's mediated by a lot of molecules or growth factors that have been shown to be important in development of the tooth. And Phil's already mentioned that we work on novel molecules such as hepatocyte growth factor, which Phil did as part of his PhD. And so there's a lot of these molecules involved that are released from the dentine matrix that can stimulate dental tissue repair. The processes that are involved are either relatively simple, a reactionary dentinogenic response, where you've got tissue damage, release of the growth factors, and they'll stimulate the odontoblast to make more dentine. So an odontoblast mediated response, so it's early stages, it's dental tissue or caries arrest potentially, so in early stages of disease or following maybe clinical intervention. Or then there's a more complex stem cell mediated response. We can see a dentine bridge. I think this is from Stefan Simon's paper, where there's been an MTA restoration, and you've got some dentine bridging, some new odontoblast-like mediated response. Odontoblast-like cells have differentiated from stem cells likely derived from the pulp. And this is reparative dentinogenesis. I'd say the two processes are distinct, both giving tertiary dentine beneath the site of the lesion. We're upregulating odontoblast-like responses on this arm. On this side, however, in the reparative dentinogenic response, we're getting new stem cells, or getting stem cells to differentiate into odontoblast-like cells, once again likely due to the growth factors that are released from the dentine itself. These odontoblast-like cells will have proliferated from the stem cell population, differentiated, and they'll upregulate mineralization generation beneath the lesion. So we've obviously worked with stem cells in Birmingham, and we look at biomimetic environments surrounding areas such as tissue engineering. And obviously you've got the link between scaffolds, stem cells, and signaling molecules. We've also looked at pulp matrix components to see if we can better understand how the dental pulp stem cells respond. So we've done studies that we published a few years ago in Journal of Dental Research, trying to recreate a biomimetic environment using pulp matrix molecules and hydrogels, alginate hydrogels. So if we isolate pulp proteins from pulpal tissue, so we use a process of protein extraction. This just shows that we could isolate these matrix proteins. We can coat them on plasticware. Some SEM shown that we've coated the plasticware, some staining to show that we put proteins there, and then we put whole pulp preparations of cells onto this. What we find is that the cells over a period of time actually don't grow much faster. They don't proliferate, but they adhere, and they sit there quite happily. So what we hypothesized is that rather than growing cells on plasticware, if we could create an environment that was more biomimetic, similar to what's occurring in pulpal tissue, maybe we could keep stem cells in a more stem cell-like state, i.e. with greater ability to differentiate. And I'll just kind of go through this relatively quickly, but if we looked at the cells that were grown on the pulp matrix, we could see that they had greater expression. Whilst they weren't proliferating very fast, they showed greater maintenance of mesenchymal and other pluripotent stem cell markers. And if we did high content analysis for OCT3/4, we also got data that supported that. If we took those matrix components, we took those cells that had been grown on that matrix and we tried to differentiate them, then we found that they differentiated better as well in this more biomimetic environment, i.e. in collaboration with the matrix proteins we'd isolated from the pulp. We took those matrix proteins, we put them into an alginate hydrogel, we put pulp cell populations into the alginate hydrogel. This was what we used to cast the alginate hydrogel, and we put that in a micro CT after 5 weeks, and we could see this kind of mineralization potentially around the edge of the construct that we'd made. And you can see that in this construct, after 5 weeks, we've got this kind of potentially laying down this opaqueness, which was perhaps suggesting mineralization was occurring around the edge. We did H&E staining, alizarin red staining for calcium, and we could see that we were aware, we've done stuff, I'm not showing it here, but we show the cells remained viable for this period of time, and there was increased kind of protein and calcium deposition around the edge of the construct. Whilst this doesn't generate a tooth that we'd want to put back into a human being, we found it was like proof of principle data to suggest that this was a nice way to generate perhaps a process of which we could test materials, for example, and study stem cell behaviours. So potentially this could all lead to, in the future, we could put hydrogels into teeth, either with cells or without cells, and try and either recruit stem cells into the hydrogel, or the stem cells that we put in the hydrogel may then differentiate, lead to a new pulpal tissue, and also in younger individuals lead to complete root formation. We've looked for new molecules as well. As Phil's mentioned, hepatocyte growth factor. We've done microarray analysis of carious compared to healthy tissue. We found lots of genes that are differentially expressed between these different populations of tissues. There's a lot of inflammation and immune response occurring. We found one molecule that we were highly interested in, a molecule called adrenomedullin. We found it was abundantly expressed in carious tissue compared to healthy tissue, both at the gene expression level and the protein level. And then when we profiled it in a whole bunch of different tissues, we also showed that it was relatively highly abundantly expressed in pulpal tissue as well, healthy pulpal tissue, compared to all these other tissues as well. We went on then to characterize it in tooth development, its effect on the proliferation of dental stem cell populations, and found this biphasic response, i.e. a positive stimulation at low concentrations, whereas the high concentrations weren't too good for the cells. So it was obviously time and concentration dependent. And we could also stimulate mineralization events with this molecule. Quite interestingly, this molecule modulates infection, or can be affect bacterial viability, i.e. kill bacteria, is bactericidal, and modulate the host inflammatory response as well. And our data and that from others have shown it's important in regeneration events. It's quite a pleiotropic cytokine. I just want to briefly touch on a couple of areas that we work on in Birmingham, just in case this is any further interest to you guys. So, low-level light therapy and photobiomodulation. I'm aware that you've got you do work in the laser area, etc., and light therapy areas. So we got into this area a few years ago. We certainly haven't got the extensive experience that you guys have got over the 20-year period, but we've done it for 5 or 6 years now, and we've had funding from the British military and NIHR, which is National Institute of Health Research, and industry in the UK. So looking at the 600 to 1,000 nanometer area, and obviously it was discovered by its ability to stimulate healing responses, or in this case regrowth of hair on the back of animals, and obviously it's been used in human systems where it can regrow hair, and it appears to be whitening this chap's teeth as well. It's not, we don't do much photodynamic therapy. It's more the direct effect of light, and as I'm sure some of you are aware, this process occurs by light being absorbed by mitochondria, activating the COX enzyme, releasing nitric oxide and stimulating reactive oxygen species. And it leads to all these positive responses in cells, cell proliferation. It's meant to be anti-inflammatory, increased cell adhesion, mineralization. We published some work a few years ago in JDR on this. We use this light therapy system to do that. And this work was mainly done by Michelle Holder, but we have clinician involvement, we have photophysics involvement as well, so it's quite a holistic team to better understand how the light's delivered and also the cellular biological responses. We show that the light stimulated proliferation, metabolic responses and energy production, and also able to stimulate mineralization in pulp cell populations. And we had this biphasic response. If you went past a certain point, you lost the positive effect. You didn't have a negative effect, but you lost its positive effect. We've gone on to generate these light therapy arrays where we can look at lots of different wavelengths, and we can look at lots of different cell types. And this is one of the areas that Sibele worked on when she came over to Birmingham. Epigenetics is an area that we're particularly interested in. Phil's also mentioned Hal Duncan, who did his PhD with us in Birmingham, but is in Dublin, in Ireland, and he's looked at the epigenetic response in pulp cells. So actually, the DNA wraps up very tightly, and if you can relax it using histone deacetylase inhibitors, you have the potential to stimulate differentiation much easier, change gene expression to enable cells to undertake repair a little bit more easily. And he's published quite a few papers from his PhD in this area as well, with myself, Tony Smith, and Gary Fleming. And basically, just to sum up this relatively quickly, if you put increasing histone deacetylase inhibitors onto pulp stem cell populations and look at mineralization processes and protein production, you can see you can stimulate mineralization processes in pulp cells and positive gene expression changes that associate with dental tissue repair. And we've characterized that in collaborations with NYU in the States as well, looking at potential mechanisms by how this might work. So just to sum up, really, in future dentistry, we believe more biological-based approaches are likely to come to the fore over the next few decades. And obviously, we need to work with dental materials to harness these processes as well. We're quite interested in dental therapies. We don't do a lot of laser work, but we do high-powered LEDs, looking at human cellular responses or eukaryotic cellular responses, and also direct antibacterial action. And as I say, we work on hydrogels, histone deacetylase inhibitors, with a view to looking for ways to increase the ability for tissue repair in the tooth. Lots of people do this work. We've got excellent technical assistance from Michelle, Gay and Helen. Lots of people working in the pulp area, perio area. We do a variety of other areas, micro CT, phototherapy, etc., etc. And the Queen opened the dental school, and I advised her at the time on how to brush her teeth. Since we've been here, we've done a bit of research, and we found that this is quite dangerous, so just be careful. Okay, thank you very much. Thank you.
Thank you very much, Dr. Bob. Let's see if it works. Camilleri. Camilleri.
Fine, whichever way you want to say it. So,
Who's this? Phils, thank you. Funcionou? Pegou o passador para ela? Camilleri.
good to see you again.
Good to see you.
It's just allergies, don't worry. It's okay. I can take a few bucks.
Oh, meu Deus. Procurando. Deixa eu ver se... deixa eu ver se... Tá bom. Deixa eu largar aqui. Mas se falar assim, você escuta bem. Ok. E agora ficou todo tonto.
Problems with USB Type-C and this optoma with back.
Every time. Never. Give it some time.
Okay.
It doesn't like PowerPoint. Okay, here we go now. Thank you very much, Professor Josette Camilleri, that is going to speak to you now. Thanks for coming. Thank you. They asked if you could speak a little louder because the air conditioning is too noisy.
I am quite loud, don't worry.
Trying to pass the... Okay, it's working.
The lights, maybe? Okay. This is good. You can have a nap this afternoon. So, good afternoon, everyone. It is so good to be here. This is my third time in Brazil. It's always a great pleasure. In fact, I'm a bit not 100% functional today. Too many caipirinhas. We've been doing caipirinhas. It's extremely dangerous, as Paul was saying, and we have, in fact, given it a new name. We call it capybaras now. It's no longer caipirinha. We decided it's a capybara. So for us in Birmingham, we drink capybaras. Actually, we had the opportunity to visit the real capybaras today. So we're getting a bit of everything. So today I will be not giving my usual MTA lecture. I've decided to diversify a bit, and I will be giving you an overview of dental material testing at the University of Birmingham. Before I start with Birmingham, I will give you a brief overview of myself. There are quite a lot of people I have never met before, so maybe you're not familiar with my work and not familiar with my research. I am originally Maltese. Again, it is lost somewhere in the Mediterranean. So when I looked at the map, I was checking. If you see all Brazil, you cannot see Malta. To be able to see Malta on the map, you have to really, really make the map grow larger and lose half of Brazil. So together they're not possible. So Malta, for who doesn't know, is south of Italy, north of Libya. It is in the middle of the Mediterranean, extremely small island. So essentially, it is just 316 kilometers area. That is extremely small. It is much smaller than Sao Paulo. Population is just 400,000 people. And when I say 400,000 people, it is very densely populated. So 400,000 people, I mean, I've seen some driving in Sao Paulo we are even do worse than Sao Paulo. Like, we just touch cars, move on, and speed. So it is very densely populated. We have a Maltese language. It is a mixture of Arabic and Italian. So even though we are so small, I call myself nowadays limited edition, because I come from a small country and I have my own language, so I am a little limited edition species. So a few images of Malta. Very nice and sunny, nice Mediterranean Sea. We go very, very back in history. So we have Neolithic temples. They are older than Stonehenge. I always remind my British colleagues that we go older than the Stonehenge, which is in the UK. It is 100% Catholic country. So all our skyline is dominated by the Catholic churches. We have a lot of fishing villages. We do a lot of fishing. We eat a lot of very nice seafood as well. A little bit about my background. I am a clinician. I graduated in 1992. I've done a PhD in materials. I am, I think, one of the few people who have started from endodontics rather than from the normal materials and prosthodontics people. I started from endo and proceeded to do a PhD in materials with Tom Pitford at King's College London. I spent a number of years doing clinical work, and all my research, I would like to think, that is based on my clinical practice. Anything I observe in the clinic, I take it to the lab. They may be small observations, but I have always managed to find something very interesting to do with my small observations. Another important thing, and I think it is also a first for many people, I spent 8 years in civil engineering. I studied all my MTA while I was doing civil engineering. I did civil engineering on a large scale, so I have published papers on wastes, concrete on a big scale, and I used to teach concrete to the civil engineering students as well. I was also an associate professor in Malta until last year, when last year fate made me meet Dr. Phil Thompson at the British Endodontic Society meeting. He said they had some positions available in Birmingham, and that is why I have moved to Birmingham. A lot of people ask why the move. I'm sure that after you've heard Dr. Thompson and Professor Cooper, you will realize that with my research, I fit in very well with the Birmingham team. I did have to purchase an umbrella and I had to change my style, but well, still now it has worked. Till now it has worked. So my research, building materials, I put it first. I still love doing building materials. The dental materials, hydraulic endodontic cements, I don't call them MTA, they are hydraulic endodontic cements. I stress that that is the term that should be used. I do some material development. I've created some prototypes. I do a lot of characterization. I stress the characterization is a very, very important aspect of materials and materials testing. We look also at tissue interactions, and nowadays I'm looking also at in vitro, in vivo correlations. My international collaborations, as you can see, Brazil is there. I've been collaborating with Brazil for over 5 years. I have to say it's been a wonderful experience. I started my collaboration with Mario Tanamoro-Filo from Araraquara. I've managed to get, today I was bragging about this, I've managed to get my 5th FAPESP scholarship. So I've managed to get FAPESP for PhD, FAPESP for postdoc, and last week I got my 5th one. So I'm very pleased. I heard it is very difficult to get FAPESP. I'm very pleased that in the last 5 years I managed to get 5. So, yes, but I get the FAPESP because I get very good Brazilian students. The FAPESP comes from Brazil, you get good students, and you manage to get the awards. So it is all about you people, but I've managed. So my collaboration started with Mario Tanamoro-Filo, then we moved with Bauru. I've had a brilliant student from Bauru. She's now in Piracicaba, and I also now have a collaboration with Fortaleza. So as you can see, I am a little bit all over the place. I've had other collaborations, a lot of collaborations with France. As you can see, civil engineering, Marseille, where there is a Madabu, I've collaborated as well. Some collaborations in Germany, Switzerland, and in Iran. Had a very good collaboration in Spain. So that is how it goes. You go to places, you meet people, you collaborate, you publish. And I have to say that when I started collaborating, my research profile has grown. The other people's profile has grown as well, and it has gone extremely well. So I do encourage everyone. I mean, my upbringing in Malta was what you know you keep to yourself. Because if you teach other people, those other people will overtake you. That is not right. It is a very, very bad attitude. What you know, you tell other people. Other people will have their ideas. You put your ideas together and you move up together. If you stay on your own, the ideas will stay with you and will die with you. So collaborations are extremely important. So, as I said, I do travel a lot and I do get the opportunities to mix with the local people. So this is this year at the European Society. I'm actually wearing the same clothes, so I should have put this off. I'm realizing now I'm wearing the same attire. So, this is Costa Rica. I do get into the natural habitat occasionally, and that's the Tucan. When you go to the Middle East, you have to dress up and you get to lecture with the imam on your head, so you have to wear a hijab, but this was hilarious, my hijab. I hadn't realized people wore black hijabs, so for me, wearing the hijab was putting a scarf around your head. So you could see me actually coming, because everybody was in black, and I was wearing all these very colorful clothes, so you could see me coming from a very long distance, but I managed. When you go to Paris, you have to dress up because it is extremely cold, but this is my favorite one. Who can tell where I am here? Oh, it's Fortaleza, of course. So I spent 7 days in Fortaleza, put on a good 5 kilos after Fortaleza, and we did eat a lot of seafood, got a suntan, very, very, very pleasant, and I also got a collaboration in Fortaleza. So it was worthwhile going to Fortaleza. Very enjoyable. I did go to Lebanon. This is Lebanon. That is a Roman temple in Lebanon. And I like resting on things, so I even managed to rest my arms also on the pyramid in Cairo. And this is myself. I believe this was lecturing in Mexico. So yes, you do go around and you meet a lot of people. So also I have edited, and I stress edited, the book Mineral Trioxide Aggregate in Dentistry. This is what I am known for, the mineral trioxide aggregate. I even got the crown. I am known as the MTA Queen. I've also contributed a chapter to Harty's Clinical Endodontics and the chapter in glass ionomers. The good thing that with this book, as you can see, there are a number of very eminent names in Brazil. And when I tell you that this book did not cause me any problems, I speak to my colleagues and they tell me they've had to chase people, they've had to run after this, that, and the other person. With this book, everybody who contributed sent things ahead of time. Very few corrections, everybody knew what they were doing. It was published on time, and we've had very good reviews about this book. As I said, I have a second family in Brazil and I really enjoy it, and we do a lot of very good work together, and this book to me is something I treasure because I've done it with my colleagues, my friends, and it is to show that these collaborations do work. So I've moved to Birmingham. I moved to Birmingham again, thanks and also the fault of Phil Thompson, and I am now located in Birmingham. You've seen Birmingham is about 1.5 hours away from London. It is very central. There's a very, very nice young community. There's a very large university, and the university contributes a lot to the Birmingham Society. So there are a lot of very, very young people around, very many places to go to. It's a nice city and a very, very young population there. This is the main campus. This is the dental school. It's a recent investment. You've heard all about it. I don't need to repeat. Again, I am not a Brummie, so I don't know much about Birmingham. This is the school. We are located on the 3rd floor. The rest is National Health Service and clinics. Courses we offer in Birmingham. We do the undergraduate BDS, which is a 5-year degree. We stress a lot of materials. I've seen a lot of people here interested in materials. In Birmingham, we are very strong in materials. So essentially, we start offering the biomedical material science as an undergraduate degree. It's a bachelor's degree. Then we have the research degrees. We have a master's in research and the PhD. But now this is brand new. I will give you some details later. This is a new master's that we do in dental material science. It is very, very holistic. We also do a couple of courses by correspondence. I will give you the details and who to contact. This is a very good opportunity to start working with places which are international places. So essentially, you can still live in Brazil or wherever and be able to do a course in collaboration with Birmingham, and you will get an MSc from a UK institution. So if you are interested in advanced general dental practice MSc by distance learning, you contact Professor Trevor Burke, an extremely well-known personality. Trevor has been all over the place and he's researched, published in restorative dentistry. These are the contacts there. If you need further details, you can contact me. You can contact Louis Mackenzie for the degree in restorative dentistry, again by distance learning. And this is our latest, our baby, our pet now, the Dental Material Science MSc, which is being run by Professor Will Palin. I know he needs no introduction in Brazil, Will. He's very well known. This is the contact details there, and these are the modules. Very, very interesting and very holistic. This is starting to be run this year, next October in 2018. So I did get a couple of shots and they weren't too happy to be there, but for who hasn't met them, these are the rest of the materials people. This is Will, this is Dick Shelton, and together with Paul and all the other parts of restorative dentistry, we do research together. This is our research team. I also have managed a lot of collaborations for which I have to thank Professor Cooper. I've been only there for 6 months, and within Birmingham, besides my international collaborations, I've managed all this. Within the first 6 months, I've got collaboration in many other places, a lot of equipment, a lot of interesting things, and it is very good to collaborate with the rest of the people at the University of Birmingham. So, my research, I will give you some more details. Dentine replacement materials, root canal sealers, root end fillers, and perforation repair, all revolving around the hydraulic cements. I am known for the characterization. A lot of people find this characterization business maybe a little bit frivolous, and you'll say, oh, but this is just some pretty pictures to present in papers. You can do a lot of work when you start characterizing. I see a lot of beautiful biological work on materials that have never been characterized, on materials we know nothing about. So I was taught always by my PhD supervisor, dentists go to the deep end immediately. You have to know your materials first. Then you start testing the material for biological properties. People seem to forget this. We characterized, and then in the discussion you start reading, we don't know why we get this behavior. It needs to be investigated. If you do characterization before you do the testing, you will know why you get a specific behavior. So it is very, very important, and characterization should not be electron microscopy only, because that is the easiest. It should follow the full characterization. It is very important that the full characterization of materials is undertaken. So this is some of my characterization work. Obviously, these are the MTAs. This is an MTA Angelus. And this is like, everybody tells me, oh, but those are great pictures. You cannot see much. You have to learn how to look at electron micrographs. With electron micrographs, I like calling them my 50 shades of gray. They are gray, but they give you a lot of detail. So this is like looking at radiographs. When you are a first year, second year student, people will push radiographs in front of you and you just nod. Yes, I'm seeing, you won't be seeing anything. I will say, yes, of course, I'm seeing loads of stuff. No, not seeing anything. But then with time, you start learning how to observe the gray and the shades of gray. So, the same with the micrographs. So, first you start observing cement particles, you start observing rims, and then you can compare that this material is different to that material. Again, this material has the shiny stuff, and with the help of other characterization methods, you will be able to tell exactly what is happening to your material. Then further characterization, and again, this was an expertise where I am proud of, because for MTA testing, everything was done using the scanning electron microscope. It was I who introduced the X-ray diffraction analysis for hydraulic material testing, because these materials, unlike the glass ionomers, are fully crystalline, so you can get very, very specific patterns, and each peak, you can see the different labeling, each peak will give you a specific phase. So if I am modifying a material by treating it in any way, I can easily follow what is going on. For example, here we do not have a calcium hydroxide peak. So I am using this material, I believe this is a terracal, I'm using this material and no calcium hydroxide is formed. And then I am very surprised that I don't find cell attachment. I am not surprised at all. No calcium hydroxide, no cell attachment. Calcium hydroxide, it behaves much better biologically. So as you can see, you can predict what is happening to the material by doing adequate material characterization. Again, we started looking even, for example, at gatta perca. I don't believe what manufacturers tell me. I apologize. There are people from Angelus here, but the manufacturers give you their own material data sheet, but it is the clinician's duty and the researcher's duty to double check what is being given. For example, when they launched the coated points, I said I want to see what is on the coated point. So very, very simple, stick it under the electron microscope and it is actually coated, and we did find the calcium and the zirconia, and some people are insisting on heating these points. These points are not to be heated, obviously, because these points have a calcium layer on them. How do you heat a calcium layer? You don't heat a calcium layer. So these points, obviously, as suggested by the manufacturer, should be used with cold compaction techniques. Again, the discoloration, we've had a lot of discoloration, a lot of research has been done in Brazil, particularly with the group in Bauru, with Marco Ungaro Duarte. We learned from characterization methods that not only the material discolors, but the tooth discolors as well. But why? So if you look at the electron micrograph, you don't get much details. This is the tooth, that is the material, but you can do mapping. So if you map and you know that calcium is this aquamarine color and the bismuth is yellow, if this is the material, this is the tooth, you can see a layer of bismuth there, which within 28 days has moved from material to the tooth. If you look at all the bismuth data, everybody says that bismuth is inert, it stays with the material, it is just a filler, it does not move. Easy. You can discredit all that easily by doing electron microscopy and scanning. Go in a scanning mode and you will find that the bismuth is there, and that is why you get all the material discoloration. So as you can see, material characterization aids your hypothesis and helps you to know what is going on in the clinical practice. Okay, we seem to have been blocked. Let's see. That's okay, it's just my computer playing up. So how do I exit from here? It's Outlook. Somebody sent me an email, I think. Okay, yes, we got rid of it, thank you. Okay. So we did the same. We did also some mapping of subcutaneous implantation. Again, I tried to have a different approach. So I know here you do a lot of animal work. So most of the times people will do subcutaneous implantation of the material and do the routine histology. In my case, I look at it from a different perspective. I do the routine material histology. However, I also retrieve the material and characterize the material after it has been implanted. Again, this approach is simple, easy, but not a lot of people adopt it. So what we have done here, and that is how we realized that the bismuth was extremely unstable. This is the implantation site, this is the skin, and this is the animal hair. This is electron microscopy, but then we did the maps, and if you look at the bismuth there, you can see that the bismuth is all over the place. So it is not only at the implantation site, but it is everywhere. I mean, as I said, simple things, but it adds a lot of spice to the research that you do. And this is what makes the research publishable, and publishable in journals that are of a higher standard. I mean, you can publish anywhere, but the higher the standard of the publication, the better for the institution, because when you undergo the ref process, obviously you will get a better rating. So these are some tips on how to push the research further. Characterization, as I said, this was my work on the discoloration, purely lab accident. So essentially, I was observing in the clinic that every time we used MTA with regen endo cases, all the teeth were discoloring. Every time we did endo, perforation, repair, the tooth discolored. So I said this has something to do with the endodontic procedure. So what I did was, I did some bucket chemistry. I had big containers. I threw cement in some containers, and in other containers I threw bismuth oxide and used reagents that are used normally in endodontics. So I said maybe because you dry it, it doesn't like the heat. This is pre-treatment. I added water, I added sodium hypochlorite, and I added also the peroxide. And this was a big surprise because I expected the peroxide to cause the discoloration, but all the discoloration happened with the sodium hypochlorite. I ruined all the benches in the Maltese Dental School because I got all the discoloration on the benches as well. But I see that here you have nice granite benches, which I have admired greatly. They're very nice and strong, and they cannot be damaged by my nasty chemicals. So that is fine. So as I said, these are simple things you observe in the clinic, which you can translate back to the lab and get them publishable. Other work that we do, which we've done, this is my collaboration with Amsterdam and also with Araraquara. We are also looking at the interface, what is now known as the mineral infiltration zone. So we can tag the materials using fluorescent dyes, and you can observe the bonding, type of bonding that is happening here. You can observe that the mineral is moving along the interface. There you get a mineral infiltration zone, and obviously the sealer tags there, which are labeled with the fluorescent microspheres. Also, something that I was observing clinically, and again, this I have worked with in collaboration with Araraquara. I had the student there. Whenever I did warm vertical compaction, as you know, AH plus is the gold standard. Every time I did warm vertical compaction, I was observing that the sealer was turning brown and it was sizzling. So you will see bubbles of sealer coming up. And sometimes when you go on social media, you actually hear them saying, these really staunch endodontists, I hated Schilderian compaction until the sealer was bubbling. And I said, something must be wrong. I looked up a little bit on epoxy resins, because epoxy resin is the base of the AH+, and I found that epoxy resins should be heated only to 60 degrees. I went to the system B, and the dial on the system B reached 200. I said, this must be a mistake, because 200 is a very high temperature. So if you think what 200 is, people don't realize how much heat we use during endo. 100 degrees is boiling point of water. So 100 degrees is something that if you throw on your patient, you'll kill the patient. 200 is double that. The only things I heat at 200 are chickens when I am roasting them. So this is being very drastic, 200 degrees. So with a little bit of clinical training, with a little bit of clinical knowledge, simple experimentation, all you need to do is simple thermocouples, and I can show you a picture of what we did. Very, very cheap, very effective. Data logger there, stick the thermocouples, have the tooth, some acrylic there, and we changed the media and measured the temperature at the external surface of the tooth. We measured even the temperature which was occurring at the tip of the system B. The system B, although the manufacturer states that it goes up to 200, it never makes it to 200. The temperature of the tip of the system B at most is 100 degrees. What we did is put our sealers in an oven for a few minutes. We decided test for 1 minute and then run simple Fourier transform infrared spectroscopy, and we found that the AH plus before heat and after heat, you will get a completely different pattern. So you see, I mean, all this is very simple research, but again translated from our clinical practice. So now I know that I should never use my AH plus if I'm using warm vertical compaction. Sealer of choice, because we did other sealers, is, for example, MTA fill latex. Before and after heat, the MTA fill latex retains the same pattern, so nothing happens there. The Septodon sealer is the bioroot. There are some changes in the water because water evaporates. Apex Plus can make the heat, but AH Plus cannot make the heat. So, as I said, things you observe on the clinic, you test them in the lab, simple techniques, and you will get a lot of results. So, from my research, I will go on to the other research that is done in the University of Birmingham, particularly into the dental school. Maybe somebody is interested in other research that we do. It's already been highlighted by Phil and Paul. We do a lot of research on caries. Recently, we got a grant and we are doing this with Oxford. We're looking at dental caries using various types of microscopy. I mean, this is very, very interesting because we've had caries forever. We think we know a lot, but really and truly we have no cure for this caries, and is still very, very, very problematic with very high caries rate in some countries. Certainly in Malta, not as much in Birmingham. They have fluoride in the water in Birmingham, but we do research on caries. We also do research on oral regenerative and rehabilitative sciences. Will not go into that, very, very well explained by my previous colleagues. Now you know how I fit in with the group in Birmingham, obviously, by looking at from the materials aspect, and the rest do the other aspects of the research. We also do a lot of light responsive technologies, again explained previously by Paul. Most of this research is run by Will Palin together with Paul and the other collaborators. Pulp, bone and mucosal tissue engineering. We test hydrogels, hard tissue repair, soft tissue repair. So again, as you can see, the research is very wide, very holistic. We do research on a lot of materials and techniques. Dental implant and bone materials, also carried out by various members of staff. If you are interested, feel free to contact us on any aspect of the research. This is my contact details. This is my email address. Normally, as the people who know me know, I reply within a couple of days, even if I am traveling. That's why this morning I was ferociously on my mobile phone, because I had something urgent to do, but I always keep myself connected. If you don't receive a reply within 2 days, feel free to recontact me. I would have missed out on the email, and make sure you get back to me. So those are my contact details. Feel free to contact me. This is Birmingham. Occasionally we do get blue skies. This is my favorite place. We have a long system of canals. I was told by a person from Birmingham, by Phil, that the canals are longer than in Venice, and I like to think that, but Professor Walmsley told me no, it is not so. But we do have a system of canals, and they do very nice boat trips. I live somewhere here in the corner. This is Brindley Place. This is the place where you go and chill. Plenty of young people there. What has amazed me with this area, this is a very, very nice posh area. What has amazed me is that all the women wear sandals. So it's soaking wet. For some reason, everybody around there is wearing sandals. So it must be a Brahmi habit to wear sandals. Very nice sandals, very colorful sandals, but all year round people wear sandals. So I thank you very much for your attention, and feel free to contact me for any queries or whatever you would like to do. Thank you very much.
Aqueles que tiverem algum tipo de pergunta, que quiser fazer pergunta para os 3 palestrantes que tivemos agora, por favor, fiquem à vontade. A gente vai fazer um pequeno intervalo depois para dar continuidade às palestras, mas quem quiser perguntar para os 3, alguma coisa para os 3 palestrantes que já passaram agora, por favor, fiquem à vontade. Sem dúvidas? Sem perguntas? Tem pergunta da sua transmissão? Pessoal envergonhado. Então, nós vamos dar um intervalo de 10 minutinhos só, e aí a gente volta com o nosso ciclo de palestras. Obrigada. A gente pode preparar já. So, vamos então dar continuidade às nossas palestras de hoje. O professor Damian Walmsley é o nosso próximo palestrante. Thank you very much for joining us in this afternoon. Thank you.
Thank you very much. And I echo what everybody else has said, and my colleagues, how much I enjoy coming to Brazil. It's my third time here, and I do like Brazilians. I've since a boy followed Manchester City, so Manchester City has lots of Brazilians in the team, and we're doing very well. So that's I like coming to with the football. Myself, and I've also. My students are watching on YouTube, so there might be some messages come through. My family are watching. They're going to get very embarrassed. But I'm a professor of restorative dentistry. I did my first degree, and then my PhD was in physics. And so a lot of my research is on ultrasonics. And I have researchers back home whose first degree is in physics and is doing research for me. Nina is my postdoc, funded by the UK government, but she comes in with a different way of bringing in and looking into dentistry. I also now spend 30 to coming up to half my time as Director of Internationalization for the medical school and the dental school. So at the moment, this is for our College of Medicine and Dentistry. The dentistry link with Brazil is very important. Next week, I'm in Singapore talking to the Duke NUS Medical School on behalf of the medical school, and Saudi Arabia to talk about cancer research with them, Melbourne to talk about interdisciplinary work. So I'm just not a dentist anymore. I cover all research from my medical colleagues as well as my dental colleagues. But this is really important to us as a university to link in with Brazil. And that pleases me because it's a country I want to link into as well. And we have been to these three institutions, and you're in the middle one and an important one. Medicine and dental sciences, we are wanting to improve our position of our league tables. As a university now with Brexit and working with Europe, we have strategies. We want to grow our research funding from international sources, so we look to international sources to grow our research funding. We want to increase our international students, and we want to increase what our undergraduate students have the opportunity to study research. And the whole university is thinking this way. We want to be for the world-class researchers, we want to be a magnet for the best students, make Birmingham a global city, and also engage our alumni. And we have several people that have come through Birmingham, say links with Brazil, for instance. So we want to influence, we want to do our research, we want to education, our resources. So this just gives you an idea of what Birmingham's thinking. We're thinking of Europe. We're looking to how we're going to deal post-Brexit. We're going to link in with Amsterdam, really strong. We have a delegation from the medical school coming that I'm arranging. And then we also link in with ACTA. We want to link in with Trinity College Dublin. We've got some German universities we want to link in with. We have a Dubai campus, which is fantastic. We have invested heavily in Dubai, and that is at the moment with computer sciences, engineering, mechanical engineering, English courses. We have a China Institute, an India Institute. In China, we look for Changzhou, the Chengdu, I've got all the things, Fudan University in Shanghai, Peking University. We see China really important, although that's not a country I'm learning about. India Institute is very strong for us for clinical trials as well, especially cancer studies. We link in with that. Brazil, we see from bioengineering, and this is from I've seen from the laboratories, very strong in your research in this area, and this is where we see we're linking in that. We also link in with Chicago, and we have links across with Melbourne, Hong Kong, Singapore, and other what we call Universitas 21 institutions across the world, in New Zealand, in Chile, in Canada, America, Johannesburg, places around the world. So we want to be up in the QS rankings. We want to increase our funding, best students, best papers. Unfortunately, everybody else wants to do the same, but we're going to do that to the best of our ability. So we want to make Birmingham a global city. Its reputation as a Midland engine. We call ourselves a Midland engine because we're the engine of the country. And we want an international reputation for our university. But that's not what I'm going to talk about. I'm going to talk about technology. And I better explain what I'm going to talk about, because in the year 2525 was, in 1969, a hit song by Zaga and Evans, and it got to #1 both in the USA and the UK. Very old song. But the lyrics of it, I think, are really exciting. And if the family are watching, they won't like this, because I'll try and sing. In the year 2525, if man is still alive, if woman can survive, they may find. How will we be teaching dentistry? And what's influencing our teaching of dentistry? And we're not just talking tomorrow, we're talking about our practicing lifetime, well, your practicing lifetime. Dental diseases will reduce. We don't think that at the moment, but they are going to reduce. People's health will get better. Then how will we teach? What will students learn? And with the politics, and what will our patients require? Now, I quite like this exhibition. I enjoy photography, and this exhibition came to Birmingham, and it was a complete immersion of virtual reality. And it was based from 1840, where they reinvented a photographic exhibition when photography was just first starting. And in this exhibition, you could pick up original photographs, and as you walked around, this is what you saw in the room. You could feel the fire. You could pick up these pictures, look at them, inspect them. Pictures that you can't actually hold because they're locked in a vault somewhere in Somerset House where they hold them, because if they get to the light, they'll just fade away. And outside, when you're in this place, there was a riot going on because there was people in Birmingham in 1840 who did not want the change in technology. Technology was taking their life away, of making clothes, of making things. They were going to have no jobs because the technology was going to change. Yet, what you actually saw was that what was I doing? In a room of nothing, I was inspecting photographs, listening. I could see mice running around the floor. I could feel the fire. So it was quite an incredible experience, that virtual reality. The first time I actually been really not just played a computer game with goggles. This was the whole experience. We got driverless cars. Maybe Sao Paulo isn't ready for driverless cars, but they're on the way. My oldest daughter, when she comes to visit us, she makes sure the heating's switched on through a mobile phone. So she uses a mobile phone to control the heating. So her house will be nice and warm when she gets back home. The electricity company gives her that. And she can put the lights on to show that people are while she's visiting us. That is a reality. To my granddaughter, Alexa, from Echo, the Echo Dot, or the Apple HomePod, it's a reality. My daughter just says, Alexa, play me The Lion King, and then she gets the music. So she talks to these things as it's just normal. So this is coming. There's people, my daughter can just pick up the phone, understands what it's doing, talk to these contraptions. And what got me thinking was when I went to an IADR conference, where Gary Slade from North Carolina talked about edentalism disappearing, that people are keeping their teeth. The idea of making complete dentures will disappear very soon. Maybe slower in some countries than others, but it's happening in some places. In London, they can't teach complete dentures because people have got their teeth. And there'll be less teachers and specialists to teach removable prosthodontics. So these are my students. This is in Stratford-upon-Avon. Shakespeare would not be too happy, maybe, to see my dental students dressed up like this. But this is near us in Birmingham. So Shakespeare is still relevant, but this is a fancy dress and shows the background and the diversity of my own students. How are we going to teach them? Are they making them ready for the future? Dental diseases will change. It's going to happen over decades. In your lifetime, it was going to happen immediately. And how will we adapt? Now, I belong to the Association of Dental Education in Europe, and we've put together in Europe ideas of what students should know about, so that it helps the schools when they're setting up and tries to put standardization across. And we're looking at dentists need to get clinical information gathering, diagnose on caries, tooth loss, periodontal disease, TMJ, cysts, tumors, cranial facial disorders, fixed and removable prosthodontic appliances, and dental implants. So they've got to do those learning outcomes. And treatment planning, must do treatment planning, successfully diagnosing the patient's condition and address the healthcare needs of that patient. And then we need to carry out the treatment, perform procedures, preserve, say, in endodontics, the vitality or all are part of the pulp. Perform procedures designed to alter the colour of the teeth. Perform non-surgical root canal of uncomplicated single root canal teeth. Now, in the UK, the government's thinking the dentistry is very, very expensive. And it's very expensive to train dentists. So they're starting to think, why don't we treat ancillary workers that can just do various bits of dentistry? And therefore, we don't have to train the dentist for 5 years. We can train people for 3 years. And this is what's going to happen to us. We have to defend ourselves why we are dentistry. And to me, dentistry is the diagnosis and the treatment planning. That's what makes us dentists. But if we give up that and just go for the mechanical side, governments will find out they can find people to do that. And the elephant in the room, we call it the elephant, the thing that no one really talks about but everyone knows wants to do. It's the elephant with implants. Everyone wants to do implants because implants will make the money. And unfortunately, it distorts everything. Distorts all our way of trying to think through the dentistry. So I think that is going to be, and we then have problems of peri-implantitis. It's not as exciting in some ways. And one of the things that's happening back in the United Kingdom is that a dentist has to pay around £10,000 a year to protect themselves from being sued. And it's because being sued because of periodontal problems on teeth and on implants. And it's getting worse in the United Kingdom, worse than America. So these are things that are the challenges that we have. And we need to be training our students, also a debate of whether we use implants at undergraduates, postgraduate, yes, and continuing education. And again, dental education in Europe is looking at that. And as people keep their teeth, 75 and over have natural teeth and with dentures. So this person is going to be wanting our treatment now, the older person. And they're going to be wanting high quality life. But they'll also have decay, they'll have a collapsing dentitions, they'll have strange problems with, they may still have to wear some dentures, dry mouths, and so gerodontology is an area that's really going to have to grow as well. So we have teachers that know best. We have students that just want to get through the course as quickly as possible and then make a life out there. But the patients getting better informed than us. They know every bit of the information as much as they can from ourselves. And also we have in the UK general dentists who tell me it wasn't like that in my day. In my day, I was trained properly as a dentist. You young people do not get the same sort of training. And that sort of distorts sometimes our way of thinking on our training. So what I want to do is just have a little look at maybe using endodontics as a way of looking at education technology and what we do in Birmingham and give you an idea. Because one of the things we are trying to do is link in with USP to look at the technology. I mean, your technology is excellent on this YouTube link. But we live in a very difficult time. Tuna fish, turbulent, the Donald Trumps of the world, uncertain, the Kims of the world in North Korea. Everything's new, but it's ambiguous. There's fake news out there. What do you believe and what don't you believe? And you want to make sure that all of us, that we're smart, we understand, and we just don't follow the line. We actually wake up and think about what we're doing, rather than accepting. That's our old dental school in 1906, when prosthetics was the main thing that people did. So our restorative course at Birmingham is specialist driven. We do initial work in the preclinical laboratory, lectures and tutorials, but we have multidisciplinary clinics. We're a school of dentistry. We're not a school with departments of endodontics, departments of prosthetics, departments of biomaterials. We're dentistry. And the reason why we're dentistry is that we can go into the university as a large grouping and get our what we need from the university for our support, for our research. We do have teaching units, but our research, everybody contributes to the research. So that's why I can work with Phil Thompson, who you would think is an endodontist. I'm a prosthodontist. But that doesn't work. That's only for teaching. In research, we link in because I have skills and links in with the engineering, mathematics. Phil's got links in with all biology. And that's how we link together, not necessarily going down the traditional lines. So I'm going to talk about what we do. And these are some of the things, our evidence-based dentistry, something called CAFs, tutorials. So we have a virtual learning environment. The university, we used to be really, really switched on our virtual learning environment, but gradually the university have locked us down so that we keep the information in just for our students. We used to have something called the e-course that won lots of national prizes, but now we put our information in something called Canvas. It works, but it's a different way of thinking for us because we were quite innovative. But we have to follow the university. So we are working through that. Now that's a lecture hall in 1906. All our lectures are recorded. Students demand that our lectures are recorded. So everything we do is recorded, and then the students can play back when they want. And why they want to record me, why they want to record me, but in fact that's what they do. They use the pause button, the forward button, the pause button, the forward button to keep looking at different points in our lectures. And all the university, every department records lectures. Now some faculty were not happy, some professors very angry about it, but now most professors, if not all, will record their lectures. In fact, while I've been away here, I probably got some of my students looking at my lectures for next week or the week after, just to check things before they go on the clinic. So a whole course of lectures has been recorded. In our tutorials, we do look to get the latest information to our students, and we not just the papers from the International Endodontics Journal, the Journal Endodontics. I quite like the British Dental Journal and something called Dental Update, which puts science findings that you might have had from your materials science, your endo science, but puts it into plain language and then is able to be read by the students, by the postgraduate students, can be read by the general practitioners. And it's a very good way of getting the science out there. The other thing is, even though we think we're great in dentistry, we have very little evidence for some of the things we do. And I've just picked on the Cochrane, and this is a blog, and there is weak to no evidence for these statements. Filling materials for treatment of failed root canals. There's no evidence which is the best one. No clinical trial has really said that. Should root canal treatment be performed in one or over several visits? There is no reliable evidence to say which is best. There's some evidence that's out there, but it's not strong evidence. Use of magnification devices for endodontic therapy. Sensible, very good idea, but nothing to suggest it makes it any better of the treatment outcomes for the patient. Because the only study that has done something has shown that people's root canals work just as well if it's been done under surgery, under microscope or not. Should you use a crown or a filling to restore a root-filled tooth? There's no evidence out there. It's interesting. No clinical evidence, yet we all have our opinions about it. Only very low quality evidence. Now, I know, and some of this is common sense, but it just makes you think that we still got a long way to go. And another one from prosthodontics is the shortened dental arch. Should those spaces be filled? On this gentleman who's 75, loving life, does he need those posterior units replacing? And most dentists will say no, but when they get hold of the patient themselves, they probably will. They'll try and make some dentures. But he's quite happy. So sometimes we forget the patient factors on it. So our School of Dentistry gives you this, that we just had a few. This spring is around the corner for us, so the daffodils will be up when I get back. We have 154 dental chairs, and we've put a lot of investment in clinical and educational IT. We have no paper records. There is no paper records. There is all our radiographs are electronic. Everything we can access on the computer here, and it's mirrored there. So every unit has got most of the units have got 2 computers. We can log in, we can write down our patient records, they're all there. Any letters are scanned in. If the radiographs come in from an outside practitioner, they're scanned in. Although it's not particularly good because the people who scan don't really think. So I find I've got pages 1, 3, 5, and 7 of a letter from a practitioner because they've only scanned the front pages of or they scan blank pages. So there's a lot we have to sometimes have a few problems with the scanning, but it does slow us down a little going electronic and IT, but the benefits is that everything is in one place. So we have no paper on our clinics. And the Queen came to visit, and this gentleman is from Jamaica, and there's a nice story about him because when he was 6, he ran after the Queen in Jamaica and met her on her travels, and now he's 70, he's in his late 70s. He dressed up to meet the Queen again in the dental school. We also have a clinical assessment and feedback system that my colleagues do. Everything about a student is on the mobile phone. So we know exactly how many endodontic procedures a student has done because they log them and we click them on the mobile phone. We know how many extractions, how many fillings, how many feedbacks from patients they've had, where they are in the year, how many times they've been to the clinic, whether their grades show that they're in the end of the year or the front of the year. And at the end of their time with us, my family are just talking to me, chatting about my lecture on here, so I'm getting instant feedback. But what they're saying is that they get a passport, And so they have an electronic passport of their time in dental school. And then they can show that electronic passport to their future employer. And their future employer can see how many fillings they've done, how much perio they've done, how many procedures, surgical procedures, where they were in the year. And you can see of this here, feedback's been provided, he'd done a root canal filling, direct restorations, he did some radiographs, root canal filling, other restorations, some direct ones. And so all these entries are done for each. They open an entry when they come on the clinic. We give them feedback, and then we close it down for them. It doesn't take that much time, but you can actually feed it back, and then you get an instant record of the student. So that's our technology. How our brave world is that we're all electronic from our clinical point of view. So it gives us real-time grading, clinical and preclinical. It's for teaching staff. We can even tell which staff are not doing their work. So you can tell which staff are falling out on the grades, which staff are giving the worst grades to the students, and are they actually in the medium? It's Big Brother. So you can find out not only what the student's doing, but whether your teaching staff are following the standards, because they might be out of sync in their standards to other people. It works on any device with a screen, and we get patient feedback. The patients, every student has an iPad, or they do it on the phone. They give the phone to the patient, and the patient puts confidential feedback in, and then they get it back. It's now being used in Hong Kong. Hong Kong, we're really excited about it, and a few other UK schools are starting to use it. We also have feedback, electronic feedback from the university. And our electronic feedback tells us how well we're doing. No, I won't close this down and get onto my PDF. But it's really good. I get really nice scores, but then I found that this year, one student didn't like me. Professor Wormsley didn't look after me properly on the clinics. But everybody else said I was great, but this one student wasn't very happy. And that sort of upset me a little, just that one. So it can be a little bit, makes you think quite a bit. One student wrote, Professor Wormsley doesn't really give much information in his lectures. I think he's trying to make me go to the library to read up about it. So I have to go to the library and read up about information. Because I give them references, I give them the bare bones, and then ask them to go to the library and to problem-based learn on the way forward. We also have links, and one of the links that I've got is I've got a Brazilian researcher, Marco Antonio Dias da Silva, and Marco and his wife are over. I think they're watching now, and Andres looks at a game technology for the students, and Marco is looking at use of short videos for as a way of getting little videos put together that the students can just dip into, say 1 minute, 2 minute videos, and then make a course up from building up the short videos. We're also looking at the web content. It's funded by the European Union. So some of you, if you were looking for postdocs, it's a way of coming into the UK. It's highly, highly competitive. But the rewards of getting one of these grants is very lucrative, not only for myself, but also for Marco, because it allows us to do the research and build the Brazilian links. So we've looked at web searching, we've looked at what's on the web, we're going to interview dental students, we're going to look at teachers, and we'll look at international partnerships. And we've also got links in with Melbourne, because someone called Rodrigo there is from Chile, so we're going to link in with Chile, Brazil, Melbourne, UK, and build in ideas of standardizations across international work. We often find that websites are not very good, and many of our teachers have problems because there's poor quality of online content, and many of the British or Irish dental courses don't have educational content that you can access. So many of our students go off to YouTube and pick up material that may not necessarily be correct, just because they find it not easy to maybe access the same material in our own university. And this is a problem. We're also looking at dental websites in Brazil. We find that the majority of content offered is in this area of the country, the Rios, the Sao Paulos, and we're looking at how the web can be used more educationally. But back to endodontics and dentistry and the biofilm we have here. Will endodontics adapt in the future? CAD-CAM endodontic instruments made for individual teeth. We can print things out, so you could just go to the clinic and print out what you need. That's not inconceivable. And robotics, it will happen. And we got together with the Americans, so the Europeans and the Americans got together and looked at what technology we'll be facing from all the teachers. And we're looking at the similarities, the personalized learning experience. We will have more use of virtual reality. Social media, curriculum mapping, lecture capture, simulcasting, what you're doing now, student response systems, laptops, tablets, phones, exam software, all becoming, no paperless, but all becoming more accessible. Simulation models, haptic technology. We're not a school that are in haptics, but we're going to Leeds Dental School is very switched on haptics and sharing their information with us. Virtual patients. Emotional simulators, so you can actually talk to somebody on a screen that will follow out a pattern for you. Big data, artificial intelligence, podcasts, progressive learning, all will change things. Patients will have showrooms. They can go in and be 3D technologies. They'll get voice operating systems. Algorithms will rule the world. You just talk to someone with an algorithm and come back with the answers for you. And so it will be very personalized telehealth. Everything will be, we will get the data. Before they come to your office, they'll have asked all the questions off the computer, and therefore you will get a printout of what you need to do for them, rather than asking them anything yourself. And holograms for patients. So how do we adapt? How do we adapt? Then we have to think more critically thinking. We must evaluate the evidence. We must be more approachable. Think of our clinical treatment planning and our case selection. Street scene in Sao Paulo. We need to personalize, make our own soup, make our own learning materials, keep them going. Think more about different years, linking the years together. Intro and intra-professional. We're doing a lot more work now of training dentists alongside medics, pharmacists, physiotherapists. And our dental students love that. There is actually something happening in Glasgow. We've sent several of our dental students there, and it's being run, and it's all different healthcare professionals. I think we need to get out of our dentistry and link in with others and be more creative and stimulate passion. Students like passion. They like people that they can link to. And it's a changing world. I have a tin of wires at home where I've kept all my old internet connections and my old phone chargers, just in case I need them for something. But I never use it. I'm going to throw that tin away when I get back home, because it's not been used. I'm always looking for the newest thing. I'm always an early adopter. So in the year 45, the song goes on to say, you ain't going to need your teeth, and you won't need your eyes. You won't find a thing to chew, and no one's going to look at you. So will we look like this, or will we be greeted like this? What will we look like? And are we an endangered species? Prosthodontics will be the first to go, then endodontics will be the next. but it won't happen for a long time, but it's round the corner. I'm very old-fashioned, so I have a Twitter account and an Instagram account, so no, I'm afraid. I'm not cutting edge like that. And thank you very much.
Agora nós vamos... Alguém quer fazer alguma pergunta para o professor Demi? Aproveita a oportunidade. O que a gente faz com os alunos agora, né? A gente estava comentando na hora do almoço que realmente é cada dia mais difícil manter a atenção de vocês, ensinar os alunos e, de fato, desenvolver novas ferramentas para interessar os alunos. E a maneira de aprender é muito diferente, né? Então, essa palestra é uma palestra que nos deixa, assim, assustados, mas, de alguma maneira, a gente tem que se preparar para esse futuro, porque ele não é futuro, né? Ele já é presente em outros países, ele já está instalado, e a gente vai ter que instalar isso aqui. Né, Flávia? Bom, agora eu queria apresentar o senhor César Bellinati, diretor de Pesquisa e Desenvolvimento da Ângelus, que vai conversar um pouquinho com a gente sobre a Ângelus e o seu desenvolvimento de materiais. Muito obrigada pela presença. Você é químico? Ele é químico, dentista? César? Você é químico? O César é engenheiro.
Boa tarde a todos. O meu objetivo aqui hoje à tarde, conversar com vocês, é mostrar como ocorre o desenvolvimento de produto numa empresa de base tecnológica e como a academia pode participar desse desenvolvimento, ou por colaboração, ou participação, ou contribuição. Mas antes de a gente entrar nesse modelo, e aí eu já vou usar uma ferramenta aqui, já mais moderna de interação. Isso aqui é o nosso fundo, onde a gente faz, que a gente chama de State Gate, que é uma metodologia. E aí eu vou navegar por essas colunas e vou explicar para vocês como é que a gente pode estar interagindo juntos. Mas antes de a gente falar do modelo propriamente dito, vamos conhecer um pouquinho mais da Ângelus. A Ângelus ela foi fundada em 1994 por um cirurgião dentista que viu uma oportunidade. É uma jovem empresa. Nós temos, então, somente 24 anos, e nós temos agora 4 focos bem definidos. A questão dos biocerâmicos, a questão de linhas de pinos e fibras, brocas carbides e a linha END. As duas primeiras, biocerâmicos e produtos de pinos e fibras de reforço, são as mais tecnológicas. Pode passar? A empresa já é reconhecida nacionalmente como uma empresa inovadora. Nós, por 2 vezes, fomos vencedores do Prêmio FINEP, uma como pequena empresa e outra como média empresa. Pode passar, por favor. E um timeline aqui, para falar mais a respeito da questão dos biocerâmicos. A Ângelus foi a empresa brasileira que trouxe essa tecnologia para o Brasil, lá em 1999. Lançaram 2 produtos na sequência, o MTA branco e o MTA cinza. Em 2010, nós pegamos um desafio bem bacana, que foi trazer o biocerâmico para dentro de uma pasta, para fazer um cimento endodôntico, e mais recentemente a gente viu que os biocerâmicos tinham desafios, como a professora Camilleri colocou, a questão do bismuto, lançou o MTA-HP, onde ele retirou o óxido de bismuto, colocou outro radiopacificador e melhorou a performance dele com plastificante. E agora, em 2017, a gente tem uma série de outros produtos que eu vou mostrar para vocês mais para frente. Mas vamos conhecer um pouquinho da equipe de P&D da Ângelus, né? Hoje, a equipe, nós temos vários mestres e doutores, nós estamos na Ângelus em torno de 80 pessoas, e que nós temos mais de 10% trabalhando em P&D. Eu sou engenheiro de formação, pode passar, Paula, por favor. Mas a gente tem na nossa equipe doutores em Química, doutores em Bioquímica, além de um grande número de cirurgiões dentistas que dão suporte para nós nesta caminhada de P&D. Pode ir? Uma outra coisa importante, né, quando a gente fala em desenvolvimento de novo produto, é a parte regulatória. Só para vocês terem uma ideia de como hoje funciona essa questão regulatória, né? Nós estamos vivendo num mundo muito globalizado. Hoje existe uma auditoria unificada, que é a MDSAP, Medical Device Simple Audit Program, que unificou todas essas ativas em relação ao desenvolvimento. Mas, pessoal, é muita norma a ser seguida e um desafio grande para quem quer enveredar e competir com uma Angelus numa aldeia global. Pode passar? Vamos aí no clinical, aqui, Paulo, isso. Bom, uma coisa muito importante quando a gente lança um produto, e aqui já começa a primeira dica de interação. Hoje a Angelus comercializa seus produtos em mais de 80 países, fortemente nos Estados Unidos, Europa, e agora a gente vai abrir esse ano um escritório no Japão. Para você vender um medical device, você tem que comprovar que ele tem desempenho, a professora Camilleri falou, uma série de ensaios necessários para você comprovar a intenção de uso, e nós, brasileiros, temos que fazer isso em relação a uma qualificação que a gente chama de qualizar. Então, aqui é um pouco mostrando de papers em relação ao MTA Angelus. Pode passar, por favor. Aqui um pouco em relação do MTA Filapex, que aí é já do cimento endodôntico, que eu mostrei naquela timeline. Pode passar. E mais recentemente em relação ao MTA HP. Todos esses materiais, se a gente fizer uma pesquisa da Angelus no PubMed, a gente colocar o nome Angelus, nós vamos ver que tem uma grande quantidade de artigos científicos se referindo aos materiais. Em relação ao MTA, tem mais de 100 artigos, em relação ao Filapex, mais de 50 artigos, e o MTA-HP, por ser mais recente, começou a aparecer artigos recentemente. Podemos passar? Bom, essa é a estrutura da empresa. Mas quando a gente vai desenvolver um produto, a primeira coisa que a gente pergunta na empresa é o seguinte: bom, qual é o problema que você vem a solucionar? Então, esse aqui está como um exemplo em relação a um biocerâmico, que é um cimento endodôntico. O problema proposto aqui é o seguinte: os biocerâmicos têm uma atividade mecânica, né, mas não exercem nada em termos químico e biológico. Essa é a problemática. Bom, diante dessa problemática, qual que é a oportunidade que se tem, então? De fazer um biocerâmico que tenha uma capacidade química e biológica como um novo material. Pode passar? Vamos ali na questão da solução ali para mim, por favor, Paulo. Bom, aqui é outra oportunidade que a academia pode participar. Normalmente, a academia ela tem um conhecimento que a gente chega com um problema, oferece para a academia, e a academia apresenta possíveis soluções de como resolver. Nesse caso, foi um desenvolvimento interno, desenvolvimento de um biocerâmico pronto para uso. E aí a gente faz uma coisa muito importante para todo e qualquer projeto, que é a parte, do escopo macro. Só clicar ali no escopo. Aqui é isso. Aqui é onde a gente vai começar a constituir o projeto. É onde a gente coloca as informações necessárias que esse produto vai ter que ter. E aí tem uma série de requisitos e atendimentos a normativas e desempenho. Podemos ir ali para a parte, não, para o último, para os requerimentos. E aqui são os requerimentos, no caso, que foram definidos para esse produto. Então, ele tem que ser pronto para uso, biocompatível, tomar presa em 2 horas, não ter pós-sensibilidade, ter uma alta alcalinidade, alta fluidez, liberar íons cálcios e ser radiopaco. Então, aqui a gente montou o que a gente quer ter como produto final. Bom, E cada uma dessas colunas, a empresa decide se ela vai a coluna adiante. Essa coluna do meio aqui, eu não vou entrar em detalhe, porque essa é a parte do pessoal da controladoria, que responde o seguinte: bom, diante da oportunidade, da solução que você propôs, se a empresa vai ganhar dinheiro ou não. Que eu costumo dizer o seguinte: nessas duas colunas é 1% de inspiração. Aqui a gente começa os 99% de transpiração. Paula, por favor, pode clicar ali no protótipo. É aqui que começa o desafio de entregar aquilo que foi proposto. Então, tem situações que a gente coloca esse desafio para a academia, tem situações que a gente faz isso internamente, e aí a academia nos auxilia na questão que a professora Camilleri falou, caracterização, para a gente provar que aquilo que a gente tinha colocado como requisito importante, a gente conseguiu cumprir. Pode voltar. Aqui é só para mostrar, ilustrar como que foi a evolução da seringa. Mas vamos entrar lá na parte de caracterização físico-química. Esse aqui já é o andar do projeto em relação àquilo que foi proposto e os atendimentos que nós temos que ter, em relação a todos aqueles critérios que nós estabelecemos. Muitas vezes a gente tem equipamento que faz isso internamente, mas tem equipamentos mais sofisticados que nós não temos, e aí então a gente pede a colaboração e faz acordos com os parceiros das universidades para conseguir. Pode ir na estabilidade acelerada? O terceiro, isso. Aqui é outro desafio de todo e qualquer produto odontológico. Ele se manter com as suas propriedades, que a gente chama de tempo de prateleira. Ou seja, a gente tem que garantir que ele seja estável ao longo, nesse caso, desse produto, ao longo de 2 anos. A gente pode fazer isso de forma acelerada, em 6 meses, ou de prateleira, permanecer com o produto na prateleira por 2 anos. Pode voltar? A questão da biocompatibilidade também é um momento que a academia pode nos auxiliar. A gente não faz biocompatibilidade interna, ou a gente contrata uma empresa ou uma universidade que nos auxilia nessa caracterização. Aqui, no atendimento, há uma norma que caracteriza isso pela norma 10933. Bom, mas, ok, então o produto ele foi caracterizado físico-químico, ele atendeu, é biocompatível, e a gente já fez protótipo. Agora a gente começa a fazer o que a gente chama de testes clínicos, para saber se o desempenho clínico está de acordo com aquilo que foi idealizado. E aqui a gente também é um outro ponto que a academia pode auxiliar, né, academias que têm os seus centros clínicos, da utilização do produto e do relato. Então, uma coisa bem bacana que aconteceu com esse produto, né, o professor aqui falou, olha, o atendimento em relação à radiopacidade tem que ser 3 milímetros da escala de alumínio. Foi o briefing que a gente deu para o pesquisador. Ele fez um cimento endodôntico 3 milímetros. Quando chegou na radiopacidade clínica, falou, olha, não, está muito ruim a radiopacidade. Então a gente alterou a radiopacidade para 7 na escala de alumínio, para que atendesse ao desejo, né, de vários clínicos que testaram o produto. Então, isso aqui foi um feedback bacana, já uma alteração em relação ao produto. E aí tem outras considerações em relação à embalagem, etc. e tal. Pode voltar? Bom, fizemos o ensaio clínico. Agora a gente vai definir quais são os critérios de aceitação que esse produto tem que ter para que ele possa ser comercializado e que o controle de qualidade possa, então, fiscalizar se isso está sendo atendido dentro da empresa. Aí também tem uma série de ensaios de difração de raio-x, de radiopacidade, etc. e tal, mas é só para mostrar como a gente internaliza essa parte importante para dentro da empresa. Okay, então, tem vários testes. Bom, a gente falou de teste clínico, mas na realidade, desde o ano passado, para a unificação da Meddev da Europa, não é aceito mais a questão de teste clínico. A gente tem que fazer uma avaliação clínica. É um documento muito mais robusto, então a gente já passou por uma série de validações e a gente vai para uma prova de fogo, que aí é uma avaliação clínica para demonstrar que o produto que foi desenvolvido tem segurança em uso. É isso aí. Ó, a gente entrar em detalhe em relação à quantidade. Eu acho que pode voltar Paula, só para a gente demonstrar a quantidade de páginas que tem o estudo clínico e quão isso é complexo. Somente depois que a gente obteve esse estudo clínico foi validado pelo órgão regulador, é que a gente vai fazer a comercialização. Eu não tenho tempo, não vou entrar em detalhe, mas depois vai ficar o contato quem tiver mais curiosidade ou interesse em debater sobre esse assunto. Aí vem a parte de instrução de uso e registro. Podemos ir para registro direto, Paulo? Obrigado. Aqui é a primeira prova de fogo, né? A gente estabelece uma premissa que o produto vai ser aprovado pelo órgão. Então, isso aqui é uma aprovação da Anvisa. Aí você clica embaixo para mim, Paula, para ir do lado. Isso, daquela setinha para a gente caminhar para o lado aqui. Isso, essa aí. Essa aqui, ó. Mais um. Mais um. Aí. Esse aqui é o documento que é encaminhado, é um documento de 300 páginas que é encaminhado para os Estados Unidos, no FDA, para ter autorização de comercialização desse produto. Então, é uma outra prova de fogo, nesse caso, esse produto já passou pela Anvisa, pela marcação CE e pelo FDA. Pode voltar? Bom, agora a empresa tem autorização para comercializar o produto. Mas uma outra coisa importante dentro da indústria é o que a gente fala de scale-up. Por favor, Paula, clica para mim ali em equipamentos. Aí eu tenho que adquirir equipamentos especiais. Pode clicar aqui para mim. E processos, no de baixo, por favor. Que garantam que eu tenha aquilo que eu consegui caracterizar e que eu consegui registrar. Pode clicar aqui, acho que tem um para frente em relação a isso. E aí, então, tem toda uma metodologia de como fazer e isso transferir para a parte produtiva. Pode voltar, por favor? Bom, pessoal, agora, depois de tudo isso, e aqui eu posso garantir para vocês que tem muito esforço, por isso que eu brinco. Aqui é 1% de inspiração, aqui é 99%. Essa aqui era proposital. Eu nem sei como é que faz isso. Bom, aí a gente tem a parte principal que é go-to-market. Mas aí a empresa falou, bom, tudo bem, eu fiz a parceria com universidades, eu fiz as caracterizações, passei pelos órgãos reguladores, mas ainda eu quero mais segurança antes de colocar esse produto no mercado. Então, a gente estabeleceu na Anjos que antes de colocar um produto no mercado, que já está autorizado, já está validado, a gente faz o que a gente chama de teste de mercado. Por favor, Paulo, ali em cima. A gente fornece o produto para uma dezena de cirurgiões dentistas que não são AA, aqui não é com relação à classe social, é amigo Ângelos. A gente pega de forma aleatória para ele dar o feedback do uso do produto na sua clínica. E ele não tem nenhuma relação com a empresa. E a gente só depois de ter uma porcentagem mínima de assertividade é que a gente coloca esse produto para comercializar. Pode votar? E depois a gente tem uma parte de treinamento, pode clicar ali no treinamento, só rapidamente. Que aí é um treinamento de toda a nossa força de vendas de como comercializar esse produto. Logicamente que é um produto bastante técnico e tem toda uma metodologia de treinamento. Pode voltar. E tudo isso condensado num technical profile. De como tudo isso que foi caracterizado, transmitido de forma clara, sintética e objetiva para o cirurgião dentista que vai utilizar o nosso produto. Para finalizar minha fala, pessoal, para não estender mais, por favor, clique no justificativa. Nós estamos lançando no mercado brasileiro uma série de novos produtos com base na tecnologia do biocerâmico. e outros tantos com base em tecnologia de fibra de vidro. Mas eu vou só me ater aos biocerâmicos, porque, na realidade, isso aqui, a minha fala é mais uma provocação. Esses produtos, todos eles, sem exceção, primeiro é um cimento selador pronto para uso, o segundo é um cimento pronto para uso reparador, o terceiro é um curativo de demora, o quarto é para fazer pupotomia em dentes tecidos, esse é para fazer restauração, cimentação, pupotomia em dentes tecidos, e esse aqui é um cimento provisório à base de biocerâmico. Todos esses produtos vão necessitar de uma caracterização mais aprofundada e uma validação. Não é fácil vender um Brasil tecnológico lá fora. Aqui a gente tem uma oportunidade, acho que única, e mais juntamente com o pessoal de Birmingham, de provar que nós realmente somos diferentes na odontologia e que nós temos uma oportunidade bastante bacana e única. Por favor. Eu estou aqui hoje, junto com a Paula, que além de operadora do PowerPoint, na realidade, é uma doutora em bioquímica, que faz parte da minha equipe. Está aqui comigo também o Roberto, que é o fundador da empresa, e a Lígia. Nós vamos ficar aqui, após as apresentações, para interagir e trocar informações e explicar um pouquinho mais de como a gente trabalha. Eu agradeço a atenção de todos. Obrigado.
Não é teu, não, né? Não. Ah, então isso aqui é para foto. É que eu apertei ali que zoou. Ah, eu vou transmitir. Não, eu vou dar conteúdo. Ah, eu vou passar agora. Você não está enxergando, né? Aqui. Não sei, não sei.
Oi, gente. Muito bom estar de volta aqui. Falei para a Adri, nem preciso me apresentar, estou em casa, né? Agradecer a presença de todo mundo. Eu sei que um monte de gente que não me conhece, porque, olha, hoje eu tive a certeza de como o tempo está passando e como eu estou ficando velho. Eu conheci as clínicas, é tudo diferente, departamento, equipamento, está tudo diferente. Né, mas está tudo muito bonito, assim, eu fico sempre feliz de voltar e ver como a faculdade evolui, né? Eu vim para cá para fazer o meu doutorado em, deixa eu ver se eu lembro, 2005, acho. Não, mais que isso. 2005. E aí eu tive um período muito feliz aqui. Então é sempre muito bom voltar. Porque eu estou representando a Universidade de Birmingham, I have to switch it to English, but it's Portuguese, so you don't need to be worried about. And my presentation is very short, and basically I'm going to talk about our team at the International Relations, because the work I do for Birmingham is this one. I am the Brazil officer, so I represent them here in Brazil, but for all areas of expertise, not only dentistry. Okay, so I will start with our team. We are these people, and we also have Erica now, because we had a little bit of change. So we have Andrea Edwards as our Director of International Development and Mobility. Then we have Catherine as International Development Manager for the Americas. So nowadays, Catherine is doing a lot of work for the US, especially with Chicago. We have a lot of partnerships with them, and also she covers Melbourne. So she gives us a lot of help when I'm there, and also for Andrea and Richard. But we also have Erica, who is responsible for the US, but she's now, I could put her here in the middle, because she's doing some work from Andrea's background and also Catherine's background. And then we have Richard, who takes care of the international partnerships, and then me. So Richard is based in Birmingham, and I'm based in Brazil, working for them overseas, and I kind of travel a little bit. And here, this slide was a lot confusing for me when I started doing this work, because here is where I am, international relations, and then I was really surprised about having all these people and all these offices giving support for our researchers there in Birmingham. And we were really happy when we've got the chance to have a FOUSP office to help the researchers with grant writing and also many other grant submissions. And in Birmingham, we have all these offices, and they take care of many, many administrative things and also support for grant submission for both staff and students. And more or less, we have an average of 25, 30 people for each office. So there's a lot of work to give support for our researchers. And here, yes, we do have a strategy for Brazil. When I started working for the University of Birmingham 6 years ago, Brazil was priority number 7, because the way we divide our work at the international office is by countries. So I was like, wow, who are the others? I'm very competitive, you know, with these type of things. And we realized, as Damien, I think he showed before, that we have a strategy with China, which is the main one. Then we have India, then we have the US, and then we had some others in the middle, and now Brazil is number 4. And after 1 year that I've got this job with them, of course, it's not my merit, but I was just learning a lot from the way they work with Brazil, and I was like, oh, let's do some more, let's do some more. And we've got, after 1 year, the Times Higher Education Award in the UK for outstanding strategies with Brazil. And as part of this strategy, FOUSP was included, and also UNESCO, that you are our main partners. So we had a little bit of visiting around. So here is our PVC International, Robin Mason. He has been doing a lot of work with Brazil, a lot of productive discussions. We've been at UNESCO. He was here at FOUSP before. We've got a previous visit from the School of Dentistry. So William Palin was here giving some lectures as well. I think last year, if I'm not wrong. So we've been at Unicamp, UNIP, FAPESP for a workshop in intellectual property. We go to a conference for internationalization to improve our work here in Brazil. We've been also talking about our partnership with FOUSP and UNESCO at the British House during the Olympic Games. So we've got a very nice partnership for one of our researchers working in forest engineering, and he has a big project now in Amazon, and we've got the content there this day. So I was really, really happy with that. And as part of our strategy with Brazil, we have a guide that we tell Brazilians when they arrive what we have been doing with Brazil. And also we have our Brazil Week. It's the second one. The Brazil Week, we have a little bit of everything. We have people talking about their previous research, their previous partnerships with Brazil, because we also have some partnerships out of the international office domain. We have some researchers that they just exchange emails, or they meet some people in conferences and they visit each other, and we just don't know. But we are trying to get the sense of how big is really our engagement with Brazil. So during the Brazil Week, we receive speakers to talk about Brazilian culture for all the international students. It's not only attended by Brazilians. We have all of them with us. We also have some workshops doing brigadeiros, caipirinhas that we call capivaras. We need to include capivaras in the program now. So it's a lot of fun and a lot of science and learning behind this. One of the things that I would like you guys to know is that we've started our engagement with Brazil in 2010, and I've started this work with them in 2011. So at that time, we have a decision to invest £2 million only for Brazil. And I think now we have a little bit more than this, because we've been doing all right. And this money goes for a lot of initiatives that we have. Some of them are, we have a visiting fellow scheme to support early researcher career researchers. So we have around 10 per year in a postdoctoral level that you can spend some time there. We have also our joint priming fund with FAPESP. We don't have any more with FAPERJ, but we had, I think, for 3 years, if I'm not wrong. But we want to start it again when they recover, and we do believe they will do. We have also supported some academic workshops. We've done some at FOUSP in the Institute of Advanced Studies. We have also some support for student mobility, and we have a scheme with FOUSP specifically for undergrads for mobility, and our PhD scholarships with CAPS that we would like to have them back. So there's a little bit of pressure from our side with them. Every time we visit Brasilia, we're like, we want them back. So, but we received many talented Brazilians over there. And we also shared an office. When I say an office, we don't have a physical presence. The office is like here, you know, and my emails. So basically, the academics are a lot busy, so I go where they are rather than they try to find me around. And we have many joint projects with Nottingham, so I cover both of them when they work together. So what do we have with USP? We have Cibele, yes. So Cibele can share a lot of experience that she had working with Paul. And this picture is here because when she got the scholarship, we've heard from Paul, oh, I need to pay you a beer fly. She's really good student. So I'm still waiting. So two things that we need to know that we have been doing with USP, and it's almost time to renew. I was not sure which place is this at USP, if it's okay, pharmacy, yeah. I was not sure, so I was like, oh, I have to ask before. So, okay, these are some projects and agreements that we have signed, but we have a general MOU with USP. So we don't need, for example, to have a specific agreement with the dental school to work with you. It's already there. It's already open. Just sit down, share ideas, and we are going to make it work. And this call was launched a month ago or 2 months ago. It's a call between the British Council and Abraham, and USP is one of the universities that is part of Abraham. And we've decided, we had many invitations, oh, we want to apply with you because the Brazilian universities, they need to find a partner in the UK. So in our case, we say, yes, we want to do it. And after many conversations and because of short time and brilliant ideas from Damien and from people from our office like Erica and other academics, Robin as well, we've decided to apply it with the University of São Paulo Dental School. And we've got it, which means that everything that you've just seen in Damien's presentation, that you're like, oh, I really would like to try this. I really would like to have it here at USP. So you're going to have a delegation of academics. Some of them are here. They're going to Birmingham for a couple of days or a week. We still don't know yet. But they're going to see all the facilities we have there. They're going to learn how we do things there. And then they're going to come back with the brain almost exploding with ideas, hopefully. And they will try to see how they could implement here all these ideas at school. And we will have a delegation, probably including Damien again, because the call is about dental technology for education. So we are going to have a delegation from there back here to follow up all these actions and give some more inputs if necessary. And also learn a lot from you, because we've seen that we have things in Birmingham that you guys don't have, but we have loads of things here, and especially we have many patients and the logistics on how we treat the patients, so we have also a lot to share with them. And we have talking a little bit more about the visiting fellow scheme, and the reason why my picture is here with 10 kilos less was because it was the first time I was in Birmingham was through this scheme. And I was, again, 3 times more when I've applied for this scheme. And basically, it's for postdoctoral level, and you have this amount of money for flights and monthly stipend, and to spend up to 3 months, and it's for all subjects. It's not only for dentistry, so it's now a lot more competitive than some years before. And I do encourage you to, sometimes we think, oh, 3 months only, but 3 months, you can do a lot of things. And it's always a way to come back. We never forget Birmingham. If you go there once, you will keep going. I always think it's my last time when I'm there, but I'm just, you know, I just can't be free of it. And we also have now the Distinguished Visiting Fellowship Program, which is a fellowship from the Institute of Advanced Studies. It's even more competitive than the other one, but we have more deadlines. So the other one we have just once a year. This one we have more. So the next deadline is 23rd of May. And then we have July, September, October, November, and you can even plan ahead for the next year. And it's for all subjects again. And for this one, for both you need to have a good CV. But for this one, because it's more competitive, you must be like an outstanding person and researcher. So some challenge, because nothing is totally perfect, some challenge that we've been facing when collaborating with Brazil is, first of all, the end of the Science Without Borders program. It was not good for them, it was not good for us here. But we are now a lot familiar with PRINT, which is the Program for Internationalization, and we know they cover some specific scholarships and strategies for specific partners. And we know we are a strong partner of the University of São Paulo, but we also know that you guys are #1. Everybody wants to do partnership with you. And you cannot just include everybody in the PRINT, okay? So we don't know even if the dental school is in the institutional FOUSP PRINT. So it's something that we could look at, because I know some of the colleges here, because I am kind of a detective when my work, I need to do investigation around what's happening at the institutions in Brazil. I know that there are some colleges here that they are approaching the rectory saying, I have this strong partnership, we've just got this grant together, we've just done this together, we have some joint publications, I usually meet these people in conferences, I want to improve our engagement with them, and then this type of approach could help us and you to be into this call, okay? So also another challenge is the industrial engagement. We know we don't have this opportunity every time to have an industry at the same time we are discussing science together. So we face this challenge because in Birmingham we have a lot of industrial partners helping us with helping them with research. So time management is also a big challenge, but I think it's more challenging for the international office when dealing with different countries and different timing for getting the responses. Lack of support from the Brazilian university, the researchers. The reason I'm putting this here is really to provoke you all, because as you have seen, our academics, they have a lot of support. And I think we need to start little by little implementing this type of thinking in Brazil and chasing the funding bodies for it. Because I've heard from someone from CAPES in a conference like this, when they are like, oh, we need to have international offices and grant support for our researchers. And the person called our researchers lazy. No, you have been studying, you have to write down everything, and you have to know everything, and you have to put all the budget together, and you have, and you have, and you have. And I think it's not only about it. I think we need help. We do need help from people that know more about business, people who know how to sell the product that you're going to have from your research. And no previous contacts, and we need the contact and to build trust. So one of the reasons why Birmingham hired me for this job was because I know a lot of people in dental school and in other schools in Brazil, and I do put people together, and I want to connect people, okay? And culture and language barriers, I think we are doing better than some years ago, but we still have these barriers. I'm almost finishing here, so we have to talk about the Marie Curie fellowships and FAPESP. We have a specific grant with FAPESP, a specific call, sorry. It's a Birmingham-Nottingham FAPESP call. Once a year, we open around April, May, and it's for pump priming research projects. So it's our auxílio regular, the category of it. And you can apply, and I know FAPESP is saying, oh, you cannot accumulate auxílios regulares, but with this one you can. So you can we can have a normal auxílio regular and then another one with us. And also for the Marie Curie, Birmingham has a really high rate of approval in these grants, and it's in a postdoctoral level or staff level. So we usually encourage people to do a pre-application around April and May. The final application is September, but why do we do this? We wanted to get it. So what happened is you write the research office, you can write me and I write them for you, and we prepared the grant with you. Not when I say we, it's the University of Birmingham, but there is an office, one of that offices specifically for it, and then together, you, the supervisor, and the officer, they work in the proposal together. And we have been very, very successful with this course, working in this way. So after the Science Without Borders, something happened in terms of memorandum of understandings and agreements in Brazil. So we've got, when they launched it, we've got a huge increase on number of agreements, and then, okay, Science Without Borders is over, we don't have any money anymore, so just some of them could keep doing some new agreements. But because of it, they thought about print. So I'm not going so much in details with print, because I think you guys are aware of it, but basically they are covering specific scholarships and some help with specific partners. And here you need to be in 5, 6, or 7 to get their eyes towards you. And it's covering the postgraduates, not the undergrads anymore, as the Science Without Borders did. And indirectly, what's going to happen is that you are going to internationalize your school, because you're going to increase the number of international students around, so you need to have websites, prospectus, CV, everything in English, and staff prepared to talk in English at the clinics and so on. And basically they cover sandwich, 6 to 12 months, senior visiting, junior visiting, capacity building, like winter and summer schools. And I know that FOUSP has a lot of good ideas in terms of winter and summer schools, so we could think about doing something like that together, because our department who takes care of summer schools, they are also really strong. And we also can have distance learning course. So from your visit this year for the British Council call, many other ideas can come up to work together. And for the staff that come here, you can offer also 15 days or a maximum of 12 months divided by 3 pairs, young talents, postdoctoral fellowships. So you can choose short-term or long-term. So it's really nice if we have a partner that you don't want to lose and you want to improve the relationship with them, to really look at the print. And the deadline is April 18. You guys as students and staff, you have nothing to do with that directly, but the rectory of FOUSP is going to submit something. And I think it's really important for you to realize what they are submitting, because it's going to affect all of you. And what they cannot cover, basically what they couldn't cover before, apart from tuition fees and bench fees, because before CAPS was covering it, and now they are not covering. So be careful when you ask for a letter to go to any university. Make sure you are telling the directors and your partner that the scholarship is not going to cover tuition fees and bench fees. And they also don't cover me coming or the similar of me here going there. So we need internal money for it. Other opportunities are BEP. You must be aware of it, as many BEPs were awarded here in the University of São Paulo. Visiting Research Program, FAPESP. We all know it's an ongoing and outstanding, and it's open for a period of 2 weeks or maximum of 12 months. So thank you very much. I would like to leave you my email. Of course, you still can find me on Facebook, on LinkedIn. On Facebook, I'm Flávia Pires Rodrigues. On LinkedIn, I'm Flávia Rodrigues, but it's the same person, the same picture, and you're going to get the same help. Okay? Thank you very much. Switch it to Portuguese again. Eu queria agradecer demais, de novo, a presença de vocês. Eu não sei se vocês têm alguma pergunta que vocês queiram fazer a respeito do que eu falei, ou que a Ângelos falou, ou mesmo alguma pergunta para os nossos acadêmicos. Queria agradecer o pessoal que assistiu a gente também da internet. Eu sei que é até mais cansativo, né, ficar na frente do computador, prestando atenção em tudo. Então, queria agradecer demais. Agradecer a professora Adriana pelo convite, professor Paulo César, Cibele, pela ajuda, todos os professores que tiveram aí com a gente conheceu a faculdade hoje cedo e depois a gente veio para essa parte de palestras, então a gente sabe que é difícil ter essa disponibilidade para gastar a manhã inteira circulando com a gente, mostrando tudo que tem aqui. Então, queria agradecer demais a todos vocês por tudo isso, tá? E com certeza a gente volta, mas a gente, antes de voltar, quer encontrar vocês lá em Birmingham, tá? E o professor Damian quer dar uma palavrinha. Obrigada.
And thank you very much from the Birmingham team for your hospitality, and also to your faculty who stopped the car from the restaurant so I could take pictures of your beautiful campus. I take lots of pictures and your campus is beautiful. You're very lucky, and I hope to visit again. Thank you.
Então, eu agradeço a presença de todos. Nós chegamos a ter 200 pessoas participando à distância da nossa transmissão. Isso é um sucesso para nós. Obrigada pela presença de todos. E vamos seguindo em frente. Muito obrigada. Um abraço. Thank you very much for coming. And I hope to see you all soon. Thanks.